and 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research, scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation.
The Honorable Dr. Laksana Trihandoko, Chairman of Indonesian Institute of Sciences, LIPI. The Honorable Deputy for Engineering Sciences, LIPI. Director of Research Center for Chemistry, LIPI. Director of Symposium on Applied Chemistry 2020 with the theme Natural Medicines and Nutraceuticals. The symposium will be held on November 18 until 19, 2020, and the symposium organized by Research Center for Chemistry, Research Center for Biotechnology, and Research Unit for Natural Product Technology, LIPI. Ladies and gentlemen, this event will try to bring up issues about natural medicines and nutraceuticals, with the topic of interest include natural product and traditional medicines, pharmaceutical chemistry, pharmaceutical technology and drug delivery system, analytical chemistry and chemical engineering, food technology, and functional foods. Ladies and gentlemen, in our symposium, there are various plenary speakers. First, Professor Dr. Renat Gunawan Indrayanto from Faculty of Pharmacy, Universitas Surabaya, Indonesia. Second, Professor Dr. Muhammad Iqbal Chowdhury from International Center for Chemical and Biochemical Sciences, University of Karachi, Pakistan. The third, Dr. Ambara Rahmat Pradipta from Department of Chemical Science and Engineering, Tokyo Institute of Technology, Japan. Niles Nirmal, PhD, from Institute of Nutrition, Mahidol University, Thailand. Associate Professor Dr. Nurul Huda from Faculty of Food Science and Nutrition, University Malaysia, Sabah. Professor Nina Artanti, PhD, from Research Center for Chemistry, LIPI, Indonesia. Ladies and gentlemen, to the COVID-19 pandemic, the symposium will conduct virtually and live from Zoom meeting room and official YouTube channel of LIPI. Of the organizing committee working with enthusiasm to prepare the symposium. In our behalf, we hope the symposium can running smoothly and all of participants would get any knowledge and making new collaboration. Well, ladies and gentlemen, please be prepared for the first agenda, National Anthem, Indonesia Raya. Ladies and gentlemen, we will start the International Symposium with warm welcome remark from the Chairman of Indonesian Institute of Sciences, LIPI, and also will be officially open our symposium. Dr. Laksana Trihandoko, the time
you all at this international conference. We hope this international conference would opportunity for all of you not only to know each other, but also to open future collaborations. I would like to thank to the distinguished invited speakers and all participants for your contribution. Have a nice, fruitful, and productive discussion during this international conference. Stay healthy. Thank you very much.
Thank you, Pak Handoko, for the remarks. Ladies and gentlemen, the next agenda, welcome speech from the Deputy for Engineering Sciences LIPI, Dr. Engineer Agus Haryono. The time is yours.
Welcome, ladies and gentlemen. Thank you to each and every one of you for being here virtually today at this important, the 6th International Symposium on Chemistry 2020. My name is Agus Haryono, Deputy Chairman for Engineering Sciences of Indonesian Institute of Sciences LIPI. It is such a great honor for me to speak on behalf of LIPI. Let me begin by giving you a warm welcome to all scientists and scholars from all over the world to take part and deliver their ideas to this virtual conference. Before we get started, I would like to express my appreciation to the honorable chief guests, respected directors, speakers, and all our other guests. We could not have done this conference without your participation. I am also grateful to all the 6 ISAC steering committee members, the organizing committee members, and all the volunteers who have arranged this program. I will kind-heartedly welcome all the participants from academia, students, industrial practitioners, and researchers. We are also fortunate enough to get supported by our partners, whom also organized this scientific conference. This conference is a part of Indonesian Science Expo 2020, aimed to bring together researchers, academians, and industry to share information, to share knowledge, and also their research results. I do hope that this conference will serve a platform to provide solutions to the problems on natural products and traditional medicines, pharmaceutical chemistry, pharmaceutical technology and drug delivery systems, analytical chemistry and chemical engineering, and also food technology and functional food. I wish you all a fruitful and productive discussion during this conference. Last but not least, I would like to sincere thanks to all of you for being able to join this virtual conference. And please stay healthy and happy during the long pandemic. Again, thank you very much and enjoy the conference.
Ladies and gentlemen, also we will hear the speech from the Chairman of International Symposium on Applied Chemistry 2020, Mr. Abdi Wirasiptama, PhD. The time is yours.
Very good morning, ladies and gentlemen. I'm Abdi Wirasiptama, Chair of ISAC 2020. Welcome to the 6th International Symposium on Applied Chemistry 2020. This conference was organized by Research Center for Chemistry, Research Center for Biotechnology, and Research Unit for Natural Product Technology, Indonesian Institute of Sciences. The aim of this conference is to gather academic, scientists, and professionals from several disciplines of applied chemistry to share the knowledge, innovation, and idea. However, the current situation prevents us to hold our flagship meeting in person due to the impact of COVID-19. And the organizing committee are committed to deliver virtually the symposium to the widest possible audience through online event. This year, the theme of ISAC is Natural Medicine and Nutraceutical, where the scientific program consists of plenary and parallel sessions with the following topics, including natural product and natural medicine, pharmaceutical chemistry, pharmaceutical technology and drug delivery system, analytical chemistry and chemical engineering, food technology, and functional food. In terms of proceeding, the acceptance of the manuscript was around 80%. 70 papers have been received in order to be presented in ISAC and will be published in IOP Conference of Material Science and Engineering Index in Scopus. All those manuscripts were selected based on international peer review with independent reviewers. We would like to thank to our distinguished guest speaker ISAC 2020.
So please all of the guests and also all of participants turn on the video. The committee will take the capture screens. Okay, 1, 2, 3. Well, once more. 1, 2, and 3. Thank you. Ladies and gentlemen, there are several announcements to all of participants. First, please have a stable internet connection during our symposium and make sure that their computer has a working camera and also audio device. The host will automatically mute the audio all of the participants. And the chat room is only available for delivering questions to the ongoing discussion in the session, requesting for changing or moving to the other breakout room in the parallel session, and also announcement delivered by the organizing committees. Dear keynote speaker, moderator, and also all of participants, please don't forget to fulfill the list of attendees that the link shared by the organizing committee in the chat room. And please properly rename your Zoom ID according to your participation category to set you up to the correct breakout room in the parallel session. For the participants that want to asking question, please state your name of institution and also the panelist name that you want to deliver the question. Well, ladies and gentlemen, from now on, we will start the plenary session 1. In the plenary session 1, we have 3 speakers. Profesor Dr. Gunawan Indrayanto from Universitas Surabaya, Indonesia, Dr. Rahmat Ambara Rahmat Pradipta from Tokyo Institute of Technology, and Professor Dr. Muhammad Iqbal Chaudhary from Karachi University, Pakistan. This plenary session will be moderated by Professor Dr. Andrea Agusta. Professor Dr. Andrea Agusta is a professor from the Center for Chemistry. He got his master's degree and PhD degree from Fukuyama University, Japan from 2001 until 2006. Now, Professor Dr. Andrea Agusta is a head of Bioprospecting Research Group in Research Center for Chemistry, and from 2006 until 2019, he was a head of Natural Product Chemistry Laboratory Research Center for Biology, LIPI. Well, ladies and gentlemen, I will give time to our moderators. Professor Dr. Andrea Agusta, please take your time.
Okay, thank you very much for the time to lead the plenary session in this morning. So I already good morning and Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, everybody. So from now until the morning, we will have a plenary session, and we have three distinguished keynote speaker is already introduced by organizing committee, but I would like to introduce more detail about the speaker one by one. We have 3 speakers, and each plenary lecture will be held about 30 minutes. And then in the end of this session, we will have also 30 minutes for discussion. So during a keynote speaker presented their works, you can ask everything via chatting rooms, or but I cannot find this. If you want to deliver a question directly, maybe you must raise your hand by a signal in this session. Okay, I would like to introduce the first keynote speaker, Professor Dr. Renat Gunawan Indrayanto. Current position is professor at Faculty of Pharmacy, Surabaya University, Surabaya, Indonesia. Latest education and training of Professor Gunawan is Dr. Renat magna cum laude in 1983 at the University of Tübingen in Germany. He ever worked as a visiting researcher at Leiden University in 1989. Also in Düsseldorf University in 1994 till 2002. Presently, his main works are analytical method development, including chemical and bioassay, and its validation, metabolite profiling, and also natural product chemistry. Professor Indrayanto currently has 120 international publications, consist of original article, short communication, and review article. His publication H 10 index were listed in Scopus, also ResearchGate, Google Scholar, Sinta, and some other database. So I would like to ask to Professor Indrayanto, already ready to deliver your presentation in this morning? Sorry. Okay.
Okay, good morning. Thank you for your nice introduction. First of all, I would like to thank to the organizing committee for the nice invitation. It is a big honor for me to share my presentation in this occasion. Distinguished participants, ladies and gentlemen, I will share my slide first. Okay. I hope you can read this slide. Can you read this slide, Professor Agustin?
Yeah, okay.
Okay, thank you. Yeah, thank you. The title of this today's presentation is The Importance of Validation Method in Herbal Drug Research. Herbal drugs, also sometimes calling botanical, or phytomedicine, or herbal medicine. Herbal-derived drugs usually consist of very complex chemical, sometimes mixture of natural products, and often containing multiple herbal or plant. It is very common a single herbal product could contain many, more than one plant or herb or extract. We know that herbal drug is very important because about 80% of population of developing world use or consume these herbal drugs. In Indonesia, traditional herbal drug that were produced in Indonesia, we call it as jamu. Jamu has been used for a long time in Indonesia to maintain good health and treat diseases. If jamu standardized jamu preparation consisted of standardized drug extract and phytopharmaceutical, but non-standardized jamu is also available. Around 75% of Indonesians also consume various types of jamu. We know that there are countless scientific publications on herbal drugs or medicinal plants, but unfortunately, most of the marketed herbal drugs in Indonesia are not yet fully scientifically established regarding their safety, efficacy, and consistency best to best. This is the reason why the application of these herbal drugs by medical doctors in Indonesia is very limited. I think it is also maybe in other countries. Many published work on herbal drugs or medicinal plants did not report their method of validation completely. This means the validity of the conclusion cannot be confirmed. As a consequence, these publications have only limited value. For developing a modern drug from herbal or plant, chemical and biological assay are always required. So the availability of validated method is very crucial. Without applying validated method, our conclusion may be wrong or biased. We know that most of the Indonesian traditional drug consists of mixture of plants and or their extract. Indonesia is also very rich on natural products, plant, animal, marine biota, and all of these natural resources can be used for the new drug development. Therefore, the resources of herbal drugs or plant or other Indonesian natural resources are very important in order to develop drug for the drug self-sufficiency in Indonesia. This is the diagram of ethnopharmacological study plan. From traditional drug application, also jamu or others, usually we can do literature review and then evaluation the literature and deciding the plant material to be tested, designing the methodology, and after that we can collect the plant. After we can get the plant, usually we do the toxicity, bioassay, safety, and also phytochemical study for preparing standardized extract and or pure compound. And then we can do some in vitro study to confirm the bioactivity, and if it is okay, and then continue to clinical study. Drug can be only marketed. Actually, drug can be only marketed if the clinical study are well okay. The same diagram have been also published. This is the same, usually from traditional drug, also jamu, preparing extract, phytochemical screening, in vitro, in vivo bioactivity study, and preparing standardized extract, and also isolating and determine the pure compound. And then after that, we usually do toxicity study of preclinical study and clinical trial. This is the same also. All drugs can be only marketed if clinical trial and all trial are okay. From that, I can make summarization of the important aspect how to develop modern drug from traditional drug. At first, we should collecting and authentication of the blood material. Can be used microscopic DNA profiling or metabolic profiling. And then predicting, determining the possible active compound or toxic compound and their chemical structure using chemical and biological method. And then determining the compound that can be used as marker for quality control purposes. determining the detection limit of the contaminant of the toxic compound. Sometimes we need to isolate the active compound to determine the structure, especially this is for unknown compound. And then we can do development of standardization of extract and stable herbal drug for marketing purposes, and then clinical study of this drug. If all is okay, then we can market the drug as I previously said. To have reliable data of characterization of the herbs or plants and phytochemical or use chemical and biological method must be validated. This is the first, how to determine the active toxic compound and its chemical structure. This is two methods. The first one is using what we call bioassay guided isolation of active compound. In this method, the raw material, the plant, will be extracted with some solvent, maybe ethanol, and we can use some method, maceration, chocolate, or solidification, and then after that, all of the extract will be tested their bioactivity. Only the active extract can be continued to fractionation with some method, and then only the active fraction can be further work to isolate the pure compound. After we get the pure compound needed to be tested, and then the chemical structure of the active compound will be determined. This is the first method, bioassay guided isolation. The second one is using combination of liquid chromatography, mass spectrum, and multivariate analysis. By this method, the active metabolite or the active compound can be directly determined without any isolation. Something like this, we have profile MS, LCMS, we have profile bioassay, and then using multivariate, we can determine which sample are active, and then using export, we can also determine which peak are active. Before we use this PGA or multivariate, we should less than 30%. Variable show more than 30% should be removed. This is the example. This is show relative tight clustering, so it's means the method is valid and can be applied for other purposes. This is the step taken for predicting chemical structure using database. At first, we should, by LCMS, we can determine the exact molecular weight based on active. Active should be confirmed by extract ion chromatogram. And then we can determine the most probable molecular formula using isotope pattern. And then we can determine and confirming fragment ion based on observed mass spectrum data and their extract ion chromatogram. After we get all this data, we can insert to some database, and we will get a predicting structure and fragment, and we should evaluate it fragment and the proposed structure with other method. For confirming the predicted chemical structure of this active peak or compound, it is nice if we have authentic standard, or we can compare with published data or other mass spectrum database. That is for known compound, but for unknown compound, usually we should isolate or synthesize the compound to confirm the structure. That is the example. We can have here, I will show you some LC mass spectrum, and from that we can get molecular ion, exact molecular ion, and we can get isotopic pattern. From that we can determine the probable molecular formula. If we have all data used by molecular ion, this formula and the fragment of MS2 we can insert into some database or one or two database, and the database will predict one or two more compound. That's why we should confirm with other database. Jadi with other database, jadi or database and we can confirm the structure using other database or publication and evaluate the database using official guideline. I will detail later. We know that all organic compounds have specific mass spectrum, its molecular ion, and its fragment. This is the guidance of the Commission Decision 2002 from European Commission. This guidance shows that the identity of a peak can be confirmed if the peak has IP at least 4, and the ratio of the fragment daughter ion fulfill this table. So for LCMS, we need 4 specific fragments because the IP from mass spectrum is 1. So for fulfill 4, we need 4. But if we have LCMS-MS, it needs only 1 precursor, IP 1, and 2 daughter ion. So the total SP also 4. But for high resolution mass spectrum, we just need 1 precursor and 1 daughter ion. But the mass accuracy should be less than 5 or 10 ppm. This is the same guidance from FDA 2015. This is the guidance. This is the difference that it describes the extract ion chromatogram retention time should be not differ 2.5% or not more than 0.5 minutes. And the criteria below is the same. If for only mass spectrum, because it is high resolution, it's just 2. 2 is with 2 times 2 is also 2, but the IP is 5. But if combined with mass spectrum and mass spectrometrum, it needs 1 precursor and 1 daughter ion. The IP is 4.5. This is the guidance. If we have IP at least 4, it means that the identity of the peak can be confirmed. So I will continue to quantitative determination of group of compound that can be used as marker for quality control. But I just only discuss the most important aspect of the validation method due to the limited time. Ideally, chemical marker for the purpose of quality assurance should represent the chemical identity profile and reflect relevant pharmacological and toxicological profile of the herbal medicine. Unfortunately, there are currently no detailed accepted current official guideline which can be used to select this chemical marker of method of cutting, number of herbs, extraction method, and formulation, fabrication, active. This all can affect the biochemical content of herbs or a medicinal plant. So it means identical herbs could have different bioactivity due to different content of their phytochemical, qualitative or quantitative. Due to variation in raw material, harvesting, extraction, and formulation, as described previously, assigning a certain phytochemical or marker could be not always applied for indicating the quality of the herbal drugs. To overcome this problem, Benson et al. In 2015 proposed a ranking system of marker that could be used for quality control purposes. That is herbal marker ranking system. Each of marker will be ranked as X 0, 1, 2, 3, 4, and 5. This is the ranking from Benson. Marker X is in. There are no bioactivity studies currently available for this compound. 0, compound activity is not related to indication of the disease interest, and no pure reference standard is available. 1 is mean compound bioactivity is indirectly related to symptom or disease, or compound is present in too low concentration. The concentration is below microgram per gram, or compound is not supported by traditional and therapeutic use of the herbs. Number 2, compound has bioactivity related to symptom of the disease. However, there is only one good quality studies to support this action. And compound is presently in low concentration between 5 to 50 microgram per gram, and the traditional or current use of this drug consistent with the bioactivity or pharmacological effect of the compound. 3 is this same, the difference only that is the same, that the concentration is higher, more than 50 microgram per gram. 3 is same with 2, the difference only the marker available in high concentration. And number 4 is same with 3, the difference that there are 2 good quality study. 2 good quality study, that is quality study about the first effect and clinical studies. And ranking 5 mean same with 4, but the compound have already bioavailable. It means they have already pharmacokinetic study of the compound. Or number 5, the compound may be toxic and they need to be screened or to comply the safety limit, especially for the compound that genotoxic or carcinogenic, that the maximum permitted level is 1.5 microgram per day. In 2017, Professor Langer from Austria described a paper regarding this matter. I will summarize. Nearly all monographs on herbal drugs or herbal drug preparation currently in European pharmacopoeia contain an obligatory assay of a certain compound or a group compound and define the minimum content. This is also described same thing in USP 43 2020 or Indonesian Herbal Pharmacopoeia 2017. Generally, they did not describe the maximum content. This is different if we see the monograph of chemical component. Chemical compound should always describe the specification ring of the product. In most cases, that obligatory is inappropriate for herbal drug evaluation, as the say constituent must be considered as purely analytical marker without correlation to their efficacy and safety. It is nice to initiate a scientific discussion in order to adapt current quality standard for herbal drug and herbal drug preparation to reflect current scientific knowledge and requirement that are appropriate for quality control purposes. It means a certain chemical marker or compound that correlated to its pharmacological action must be determined and specified. This is important for quality tool. So indirectly, Professor Langer confirmed the work of Ben Susan in 2015. In 2017, this is the guidance of WHO according to marker. Describe something like this. Marker for quantification should be represent the main therapeutic or pharmacological profile of the herbal material and or finished product. Marker for identification should be specific for one plant or for certain plant, species, or genera. If it's not, other markers should be selected for specific identification. That should be detectable and quantifiable in the product. Different substance must be selected for the same herbal material depending on the different form of the herbal preparation. Maybe this is aqueous or alcoholic or tablet, etc. or different therapeutic indication. A group of substance may be selected if one is not sufficient to evaluate the herbal marker of finished herbal product. This is the guardian of the WHO. In 2017 also, some researchers describe what they call quality marker. Quality marker can be defined in the following condition. Quality markers should be existing in herbs, extract, herbal product, formulation, etc. Quality marker could be analyzed using qualitative and quantitative approach. It can be issued the authenticity or for differentiating the quality of the same herbs. The drug effect, such effectiveness and safety should be demonstrated to be associated with their quality marker. The quality marker is chemical substance that is traceable in whole process of production and preparation of the herbal drugs. So if we read this all key marker and guidance of the WHO, key marker is about the same meaning to herbs marker ranking system 3, 4, and 5 of WHO or WHO guideline. So only key marker or herbs marker ranking system 3 to 5 can be applied as a visi tool for evaluating the quality of herbal drugs. Unfortunately, quality marker is not yet described by current pharmacopoeia, and only a few quality markers have been determined and published. If quality marker is not yet specified, it is recommended to apply metabolic profiling combined with multivariate analysis as a visi tool. For having reliable data, the method of analysis of the key marker should be first validated. Its selectivity, linearity, accuracy, precision, detection limit, quantization limit, etc. And that is the summarization of the recent development of chemical marker and metabolic profiling. If you're interested, you can send email to me. I will send you the full paper of this two review article. I will choose the most important. This is the first as the selectivity. As I said previously, due to the complex nature of herbs, chromatography is the method of choice for its method of analysis. To prove the selectivity, the target peak, this target peak should be proved regarding its identity and purity, because identical retention time does not mean identical compound. One peak does not mean a single compound, so we should use direct ray detector or mass spectrum to confirm this peak. But the problem is different compounds maybe have identical UV vis spectra. So mass spectrum is better detector, but mass spectrum is the best, as I described previously. So the second important is how to express the linearity. Usually, to express the linearity, we using correlation coefficient. But correlation coefficient cannot be used anymore as whole parameter. As you see in this slide, correlation coefficient R square 1, almost 1, the error could be 1%, R square 0.987, the error could be 4%. So the correlation coefficient can be used as a sole parameter for evaluating the linearity. Actually, this has been published in 8080 by Association of Chemical Analysts of USA. So for that purpose, we should use other parameter. That is some parameter that can be used to express the linearity. The easiest one is use correlation factor, and the ANOVA test. ANOVA test can be performed by Excel. So it means that correlation coefficient, if the calibration is okay, correlation coefficient is okay. But the correlation coefficient is okay, it does not mean the calibration is okay. Sometimes if we work with natural product chemistry, the standard is very expensive. That's why some researchers propose what they call quantitative analysis of multi-component by single marker. For doing this, we should calculate the average relative correction factor. And if we have average relative correction factor, we can determine any compound with other common internal standard. But before we can use this equation, we should do linearity of all target compound, and common reference standard must be evaluated, and identification of the target peak is performed using relative detection time. This is the criteria if we want to use this equation. So by this method, we can determine any marker, any compound without using other internal standard as more cheaper. Because in many on its standard, and the difference is statistically significant. So the total polyphenol and flavonoid in the sample must be determined according to the standard that is found the most abundant in that sample. That is why selecting the correct standard is crucial. We cannot use any standard for determining the polyphenol or flavonoid. We should know what is the most abundant compound in that sample. And of course, the method should be validated. So I will continue.
Sorry, Prof. You have 2 more minutes.
2 more minutes.
Go on.
Okay, and then 2 more minutes. This is the method of how to do the limit of detection using USB 43. This is how to make the sample detection limit, and this is how to validate the biological assay. For the complete discussion, you can read our chapter. The important thing is all cells should be first authenticated, and the stability of sample should be evaluated. This is the cell that can be used, not all cells, but certain cells according to certain gradients. So this is usually something like this, and we should determine the Z factor. The Z factor. This is the equation of Z factor. So the Z factor should be not less than 0.5. So the system cannot be used. So other methods should be used. Many publications that reported bioactivity, they do not report this validation. So it means the validity of the data can be not confirmed. So that this may be biased or wrong. This is also should be determined the selective index. Selective index should be as high as possible. If selective index is below 1, it means that it's not acceptable, although the compound or the extract is very active. For continuing the work, we should do accurate potency of the test sample must be determined. So I recommend use the European Pharmacopoeia 2020 section 5.3. That is better. And this is the conclusion. Indonesia is very rich of traditional herbs and alternative resources that can be developed into phytopharmaceutical or modern drug. It is recommended to determine the quality marker and its specification string for each of herb monograph as described in Pharmacopoeia Herbal Indonesia. Performing clinical trial for all marketed phytopharmaceutical is needed to have consistent quality of every batch of the product. For having reliable conclusion, all these chemical and biological assay should be validated. So, due to the limited funding and facility, I recommend we do some collaboration between all scientists in Indonesia to save the funding and to use the facility that we do not have. At last, I would like to acknowledge to Dr. Abdi Wira Saptama for the nice invitation, and thank you for your nice attention. Sorry, I will present at last of my presentation in very fast because the time is limited. Thank you very much.
Okay, thank you very much, Professor Gunawan. We have already saw the prominent result of your work concerning herbal medicine. You already present the one most important aspect in herbal medicine development, especially for quality control of quality of traditional herbal medicine. Okay, audience, we will have discussion in the end of this plenary session. Please keep your question until the discussion session. Okay, we go to the keynote speaker too, Dr. Ambara Rahmat Pradipta. Already joined in this room? Okay. Dr. Ambara, current position is assistant professor in the Tokyo Institute of Technology from 2006 and 2008. And latest education and training, graduate from Osaka University, Graduate School of Science, Department of Chemistry for PhD degree, and also conduct the postdoctoral researcher at Osaka University in 2011 and 2012, and also postdoctoral research in RICAN from 2012 and 2017. Presently, his main works are bioorganic chemistry, biorelated chemistry, natural product organic chemistry, organic synthetic chemistry, and also organic analytical chemistry. Dr. Ambara currently has more than 50 international publications consisted of original article, short communication, and review articles. He also became a principal investigator and co-investigator for several research grants. In this morning, Dr. Ambara will present the presentation titled Reactivity of Acrolein Released from Cancer Cells. Okay, please take your time, Dr. Ambara, for 30 minutes. Time is yours.
Okay, thank you very much. Good morning, everyone. So thank you for the organizer for inviting me to this symposium. It's my great pleasure to join all the meeting here. And also, actually, this is my first formal interactions with LIPI, so I'm very happy to be here. So first of all, let me share. Okay, so I would like to present about the reactivity of acrolein released from cancer cells. So, okay. So even though the moderator already introduced my background, so I just want to mention a little bit about my research background. So until 2020, I worked in WICANG. So it's similar like LIPI in Indonesia. Actually, it's the largest research institute in Japan. And actually, during my stay.
Sorry, please unmute your voice. Sorry, Dr. Ambara. Please unmute your voice.
Okay, I'm sorry. My apologies. So let me stop. Okay. So good morning, everyone. I apologize for the technical problem. So first of all, I would like to thank the organizer and the moderators for the kind invitations. It's my great pleasure to be here. Actually, this is my first interaction with LIPI, a formal interaction with LIPI, and I'm very happy to be here. And so today I would like to introduce my recent work on the reactivity of acrolein, which is released from the cancer cells. So although the moderator already introduced about my background, so I just want to mention a little bit about my previous work. So from 2012 until 2000, I worked at RIKENG. So RIKENG is actually similar to LIPI in Indonesia. It's the largest research institute in Japan. So actually, there's a very good facilities for the chemists. Actually, for example, in RIKENG, we have a large NMR facilities. If you can see here, in one cube here, we have about 10 NMRs. So if you see these 10 cubes and this large room, we have about 125 NMR which can be used by the researchers. And also, start from this April, I start to work in Tokyo Institute of Technology as an assistant professor. So during my graduate study in Osaka University, I work to isolate and synthesize this kind of peptidoglycan fragment from the bacterial spermaton. So by training, actually, I'm natural product chemist and also I'm a synthetic chemist. So I synthesize these kind of molecules. And then when I come to RIKENG, the first half of my time in RIKENG, I used to work on the synthesis of these immune molecules, of these also in these heterocyclic molecules, the chiral heterocyclic molecules, and confirm their bioactivities. And at the second half of the RIKENG, I start to have an interest to do the interdisciplinary research. So since my background is synthetic chemistry and also the natural product chemistry, I start to think, why don't I also I do the bio-related chemistry and bioorganic chemistry. And then when I start to work on this area, I also found that my research actually can be used together with the medical doctor for the diagnostic and therapy. So today I would like to talk about this diagnostic and therapy based on our synthesis chemistry. So to begin my talk, I would like to show about the breast cancers. So you see that breast cancer is the most common cancer in women. Around 1.3 million women worldwide affected by the breast cancers. Also in Japan and also in many advanced countries, actually 1 in every 11 women affected by breast cancer at certain point during their lifetime. So if you talk about the breast cancers, there are several types of breast cancers. For example, but the most common one is DCIS, the ductal carcinoma in situ, and IDC, the invasive ductal carcinoma. So the difference between these two is that if we compare to the normal breast gland, so this is the milk duct located at these positions. So if you see the normal breast glands, we could see the inside like this. But if you see the ductal carcinoma in situ, you have the cancer cells around inside the duct, like this. But in the case of invasive ductal carcinoma, the cancer cells is already spread outside and affected other area. So the IDC is more difficult to treat rather than the DCIS. So there are several ways to treat cancer, including the radiation therapy, chemotherapy, hormone therapy, and also the surgery. In the case of surgery, there are 2 options to perform the surgery. That is the breast conserving surgery or the breast removal surgery. So in the case of breast conserving surgery, during the surgery, only the affected area removed. On the other hand, on the breast removal surgery, even though only small amount of cancer appear, but the whole part of the breast will be removed. So of course, this breast removal surgery is difficult for the patients because they will lose part of their bodies. But also to perform the breast conserving surgery is also difficult because it's sometimes difficult for the doctor to determine which is the normal tissue and which is the cancer tissues. So if during the surgery some tissue is still remaining, then it will become large again, and it will become the big problem in the future. So during the breast conserving surgery, usually the surgeon will remove some affected areas, and then he will cut this removed area into 4 pieces, and then give this to the pathologist to be analyzed. So the pathologist will analyze these tissues by using the HE analysis. It's a very conservative analysis. So by looking at the morphology of the tissues, these pathologists will determine whether it's normal cells or whether it's cancer cells. And then if it still contains the cancer cells, then the pathologist will talk to the surgeon and tell them to perform again the surgery. So these cycles perform 2 or 3 times, and one cycles need about 40 minutes. So if you can imagine, if this cycle needs to be performed 2 or 3 times, maybe it takes 2 or 3 hours. And during that time, the patient is still stay in the operation room with the surgery cut still open. So it's very long process and very difficult process. Also, the pathologist is not always available in every big hospital. It's only available in big hospitals. So to overcome this problem, many scientists are trying to develop a new method. For example, this margin probe device. This margin probe device could detect the electromagnetic wave of cancers. However, it has a 53% of false positive rate. So sometimes it's not cancer, but it's just detected as a cancer. So it's still not a good method to detect the cancers. And also we know that the micro CT or the MRI also could be available to detect the cancer. But this method only detect the presence of cancers and normals, but cannot distinguish the type of the cancers. Also, the chemists also have developed a chemical probe. For example, some groups in Tokyo University has developed this probe, which when they add this probe to this breast cancer stump, only they will react with the enzyme which present on the surface of the tissues, and then release these actions to give this triazolene, which the double bond isomerize into these positions, and also the oxidized product. So the reactions is selective, and it's also stable in physiological conditions, and the azide also it could be performed in this physiological condition, and the azide itself is quite stable. So to show you in more detail, so the azide only react with the acrolein, but with the same compound which has substituent at the beta positions, alpha positions, or the other analog, the azide does not react under the similar conditions. That means that it's only reacts specifically with the acrolein. Also, it could be performed in under the physiological conditions, even when we use water as the solvent. And the azide is quite stable under the oxidative and reductive conditions as shown here. So based on these reactions, when we add the fluorophore at these positions, the azide does not contain the acroleins. So speaking about the cancer cells, actually the cancer also has a high level of reactive oxygen species. This high level of reactive oxygen species could induce the lipid peroxidations at the surface of the cancer. So it will produce various type of aldehyde. So we could find on the literature that this type of aldehyde has been reported to be produced on the cancer cells. However, in the case of acrolein, many publications and review articles says that the acrolein actually presents in the cancer cells, but actually no experimental data for this. So by using our probe, we believe that we could give the experimental data that the acrolein actually induce produce in the cancer cell in high level. So we add our probe into various type of normal cells and also into various type of cancer cells. And after several processes, we could just distinguish them by the fluorescent intensity. So if you look at the normal cells, the fluorescence intensity is very low. But in the case of cancer cells, after the reaction with the acrolein, the fluorescent intensity is, you can see that the intensity on the fluorescent intensity on the cancer cell is higher. So to make it easier to be compared, so I made the graph here. So on the left side, this is the normal cells. On the right side, this is the human cancer cells. So the higher the fluorescent intensity means the higher the acrolein level on the cells. So you can see in the various type of cells, the various type of cancer cells, the acrolein level is higher compared to the normal cells. Also, these on the right side, all of this type is the breast cancer cells, and they have a higher level of acrolein compared to the normal cells. And also, at this point, we use the TAMRA as the fluorescence group here. We found that this molecule is the best molecules for our probe because when we just change the fluorophore into different type of react with the acrolein from the cells to produce the triazolene, and it will give the diazo compound. So because the low intracellular pH conditions, it will easily to produce a diazo compound so that it could immediately react. So this diazo compound is very reactive. It could produce carbon immediately, and so that could react immediately with the nearest organelle. So they make the covalent bond. So that's the reason why we could visualize the cells, visualize the fluorescence on the cells. And this is, we make more details, we observe in more details that the fluorescence, that the probe when go inside the cells, when they not react with the acrolein, will just go outside, but when they meet the acrolein, they will react and they just attach covalently with the organelle. And so we know the mechanism and we observe that it's specific with the acrolein from the cancer cells. So we just, what we think that maybe our probe also will work with the real tissues from the cancer. So we worked with the breast cancer surgeon. So they are one of the best cancer surgeons in Japan. They're from the Osaka University Hospital. And they gave us the real sample from the breast cancer patients. And when we add our probe to these samples, and after wash with the buffer and observe under fluorescence, we found that we have a lot of pictures actually, but I will show you only the representative data. So in the case of normal tissues, we only found the blue colors, which come from the nuclei that visualized by the Hickes reagent. But in the case of the cancer tissues, we found the red fluorophore, which come from the acrolein that react from that visualized by our probe. So we collect all the data and make the statistic on this graph here. In the case of normal tissues, the intensity is very low, but in the case of cancer tissues, the intensity is quite high. So in this case, we could observe the difference between the normal and the cancer tissues. But this is not enough because we need to differentiate these two types of cancer tissues. So we just observe in more detail their morphologies. So under the 200 times of magnifications, we could see that the morphologies can be observed by our probe. So you can see that these normal cells and these 3 types of cancer tissues cells has a different morphologies. So we can differentiate these type of tissues. And when we compare to this classical H&E analysis method, they give the similar result. The difference is that this method needs about 40 minutes to be performed. Our method only needs about 5 minutes to be performed. And also we ask the pathologists to perform the analysis. We ask them to do the blind analysis and compare our method with the classical method, and they found that the result is quite similar. So to conclude the first part, so I should mention one more time that by only looking at the fluorescent intensity, the normal tissues can be differentiated by the fluorescent intensity with the cancer tissue. But when we want to differentiate these 2 types of cancer tissues and these 2 types of normal tissue, we need to see the morphologies. And only so far, only our method, our probe, which can be used to visualize the morphologies other than the classical method. But our method can be performed only within 5 minutes. So this is the molecules that we showed on the previous slides. It's the first generations. We use on about 20 micromolars of the tissue of the concentrations, and we now have the second generations by adding this isopropyl group, the bulky group here. The intensity can be increased, so we only need 2 micromolars. And this method has been used in the hospital in Osaka University, and it will be used in all over Japan in the near futures. And we just did the press release conference at the Mambuka's office, and I think it will be soon. We want to use it also in outside Japan. So that's the result for the synthetic. I named this method as the synthetic chemodiagnosis, and also the rest, the remaining time I will talk about this synthetic chemotherapy. So previously I mentioned that by adding this isopropyl group here, the bulky group, this azide, it has a higher reactivity compared to this simple azide. So we just, to prove it experimentally, we just mix this simple azide and this bulky azide and react them with the excess amount of the acrolein. And we found that only this bulky azide could react with the acrolein, but we could not find the product from this simple azide. It means that this bulky azide has the higher reactivity compared to the simple azide. To explain this phenomena, we performed the DFT calculations. By performing DFT calculation, actually it's more reactive compared to the simple azides. Also, when we look into more detail, interestingly, this bulky azide could react with the acrolein to give this triazoline, and it's react further to give this triazole compound and also this diazole compound. This diazole compound is quite stable because it's located at the alpha positions of the carbonyl. Even we could isolate this compound, and also previous report also mention about this alpha diazole carbonyl compound. So this is actually interesting phenomena because when I think we could use this compound as a therapeutic method, which I will show you on the next slides. So if I design this compound, the drug with the carbamate link to this bulky azide, Usually some drugs have the amine group, which when it's alkylated, the toxicity is reduced. So this compound, if they just add it into the normal cells, they will not show any cytotoxicity. But when they was added to the cancer cells, which has a lot of acrolein, they will react to produce this diazo compound. And if you see these lone pair electrons, it will push the conjugate bond to release the drugs and give the active drugs only at the specific cancer cells area, or cancer area. So it will kill the cancers here. So we would like to do these reactions not only on the cells, but on the xenograft mouse. So our laboratory has the ability to prepare this xenograft mouse. So we would like to just prepare the cancer on the mouse. So it means that they have a lot of acrolein now on the cancer area. And when we add our prodrug, the reactions will, they will react with the acrolein on the cancers to give this diazo compound, and then the drug will be released only at the cancer area without affecting another tissues. So as the proof of concept, we start with the coumarin, the fluorophore here. As you know that when we add alkyl group at the coumarin, the fluorescence will be reduced. But when this, which means that this, our azide only reacts specifically with the acrolein. So we just add our probe, our prodrug here to the cancer cells and also to the normal cells. If they react with the acrolein, the coumarin will be released and we could see the increase of the fluorescent intensity. And the result is shown here. When we add to the normal cells, the fluorescent intensity does not increase, means that no reaction occur. But when we add to the cancer cells, you can see that the fluorescent intensity gradually increase, which means that the coumarin gradually release after the reactions with the acrolein. So we just change now the fluorescence with the drug. We use the mitomycin C. So this drug, when we alkylate it at these positions, they shows no toxicity. But after the reaction with the acrolein, the drug will be released to show the cytotoxicity. So at the cytotoxicity test, we found that this mitomycin could have the cytotoxicity, non-selective cytotoxicity against normal cells and also the cancer cells. But when we perform the cytotoxicity test on our prodrug, the normal cells doesn't show any cytotoxicity. But the cancer cells, since it has a lot of acroleins, so our prodrug reacts with the acrolein, and they found that this mitomycin release the free drugs to kill the cancer cells. So now we are performing our experiment on the real, but on the cancer xenograft mouse. So I just show you the result here. So we just add our prodrug to the cancer. Since it has a lot of if you see this is the control experiment. If the azide does not have any drug here, after 14 days, the tumor is increasing. When we add mitomycin, our experimental drug, after 14 days, the tumor is gone. But you can see that the inflammations occur everywhere on the mouse. It means that the adverse drug reactions in the off-target is occurring, so it's not selective. And the mouse is just have to sacrifice the mouse is dead after about 18 days. But when we add our prodrugs, we can see that after 14 days, the cancer is after 14 days. We can suppress the tumor growth, and also the mouse is could survive even more than 50 days. In this case, we injected our prodrug directly to the tumor. Also, in a different experiment, we injected our drug 18 days intravenously. But when we add our prodrug, you can see that after 14 days, the cancer is
after 10 days,
we can suppress the tumor growth. And also the
cannot be
mouse
suppressed.
Could survive
In the case
more
of
than
drug,
50
tumor
days.
can be suppressed, but
In this
the
case,
inflammation
we
occurs.
injected
So it is
our
very
product
toxic to the
directly
mouse.
to the
But
tumor.
on our product,
Also,
the tumor can be
we-
suppressed.
in different
and
experiments,
also no inflammations.
we
and the
injected our
survival
drug
rates also
intravenously to the
high
tail of the mouse. You can see that
with
my
good
talk.
results.
So we use
And
just the simple reactions of the
I
azide
would like
and
to
acrolein, and
introduce
we apply
our
this
laboratory
into the
teams.
synthetic
So,
hemodiagnosis
currently,
for
I'm
the applications
assistant
in
professors of the Professor Katsuna
diagnosis
Tanaka
of the
Laboratory,
breast cancer
and
patients.
he has given me a
And
lot of
we also
freedoms
have
to do the
performed
work.
this
And
synthetic
now I
chemotherapy
work with these
for
three
the
students, and
therapy of
also
cancers. Now on
this
the
one
animal levels,
lady,
we have
Penny-san,
performed
will go
with the
to
good
LIPI, I
results.
think this
And
next year, and he
finally,
will
I
start
would
the
like
new work
to
there.
introduce
And also,
our
I'm very
laboratory
grateful
team. So
for the Kakenhi,
currently
the
I'm
very generous
the assistant professor
research
of the
grant from the
Professor
Japanese
Katsunari
government.
Tanaka Laboratory,
So,
and he has given me a
that's
lot
all
of
my
freedoms
talk,
to
and I
do
would
the
like to
work.
thanks for your
And now
kind
I work
attention.
with these three
Thank you very
students.
much.
And also,
Okay, thank
this
you very much
one lady,
for nice
Penny-san,
and
will
important
go to LIPI, I
presentation,
think
Dr.
this
Ambala.
next year, and
So
he will
we
start the new work there. And also, I'm
we
very
still have
grateful
one more
for the Kakenhi,
keynote
the
speaker,
very generous
Professor
research
Chaudhary
grant from the Japanese
from
government.
Pakistan.
So
But
that's all
Professor
of
Chaudhary
my talk,
will
and I
be
would like to
able to
thanks
join
for
with
your
us
kind
at
attention.
10:30.
Thank you very much.
So now,
Okay, thank you very
still,
much for nice
10.
and important
So we have
presentation,
about
Dr. Ambara.
30
So
minutes
we
before Professor Chaudhary
still have
join.
one more keynote speaker,
We
Professor
will give
Chaudhary
opportunity
from Pakistan.
again
But
to
Professor
Gunawan
Chaudhary
to
will be
complete
able to join with
his
us at
presentation.
10:30.
Yeah, as
So
we
now
know,
still
last part of his
10.
presentation.
So we have
delivered,
about
so fast. So
30 minutes
please,
before
Dr. Gunawan, keep
Professor
slow to
Chaudhary
explain
join.
important of your
We
result. So
will
maybe
give
if we have
opportunity
if we still
again
have time before
to Professor
Chaudhary,
Gunawan
join
to
with
complete
us, maybe
his presentation. Yeah, as we know,
presenter
the
will
last
ask,
part of his
will
presentation
answer some
delivered
question if
so
we have
fast. So please,
time.
Dr.
Okay,
Gunawan,
please,
keep
Professor Gunawan,
slow to
deliver
explain
and complete your
important of your
presentation.
result. So maybe if we have, if we still
Hello?
have time before.
Okay, please
Hello?
unmute your.
Can you hear?
Yeah, okay.
Yeah, thank you.
Okay,
Thank you for your time. I will share first the screen, the slide.
okay.
Can you see the slide already?
Not yet, not yet. Maybe need.
Can you see the slide?
Yes, I can see.
Okay.
Okay. So for confirm that the herbal, we should determine the detection limit or quantification limit. Negative means the concentration is below the detection limit, and the detection limit must below maximum permitted limit. All methods of determination of 10 and the acceptance criteria of 1 to 4 have been described by USP Herbal Medicine Compendium. It is free. You can open this link, and all this method can be described in detail. Determination of the detection in this method of the USP 43 with a very low detection with very low concentration. The low concentration can be used by signal to noise ratio. It's mean calculate for detection limit is 3 to 1, for quantification 10 to 1. So we can start with this concentration and we can calculate the limit of detection with this equation. The sample detection limit. The limit of detection, this is the equation, and we can calculate the limit of quantification. They replace this one, by 10.
Concentration of limit of
This is
quantification,
how to calculate the detector's limit of
7
the
replication,
standard, pure
and then
standard.
determine
But
the
if we
content,
use sample,
and determine
any
the
sample,
standard deviation.
leaves, root, or
By this
etc.,
method, the
we should
detection limit
determine
can be
the sample
calculated with this
detection
equation,
limit
and
according
the
to this
quantification limit
regulation
can be
of
calculated
FDA.
with this equation.
For this
So it's
purpose,
mean
we should
for every
spike
matrix,
the
it
matrix
could be the
with
detection
the
limit is different. So that
concentration
is why
of
the
limit
detection limit
of
must be
quantification
performed for each
with,
of matrix. The
7
detection limit in
application,
blood may be
and
different
then
from urine,
determine
or
the
maybe
content
different from
and
other
determine
matrix.
the
So
standard
if we work
deviation.
with leaves,
By this
and we
method,
have with
the
work
detection
with
limit
fruit,
can be
so maybe the detection limit is different. So
So
it
it's
is
mean
important
for
to
every
determine
matrix,
what
it
they
could
call
be the
sample
detection limit is
detection
different.
limit.
So that is why the detection
After we
limit
get
must be
sample
performed
detection
for each
limit,
of matrix.
we should
The detection
verify
limit in
or
blood may
confirm
be
the
different
data
from urine
by
or maybe
determining
different
what we
from
call
other
cut-off
matrix.
value.
So if we work with
This
leaves
cut-off value can
and we
be
have work
calculated
with
by
fruit,
using
maybe
this curve. This is the
the detection
concentration,
limit is
and
different.
this is
So
the
it is
positive
important
result,
to determine
positive result
what
of
they call
the
sample
detection.
detection
For the
limit.
example, this is the detection limit.
After we get
about,
sample detection
80
limit,
microgram
we
per
should
liter.
verify
The
or
maximum
confirm
permitted
the
level
data
is about
by determining what they call
125
cut-off
microgram per
value.
liter. So
This
by
cut-off value can
doing
be
experiment,
calculated by
it is
using
nice
concentration, and
and
this
we
is
can
the
calculate the
positive result
percent
of
of positive
the detection.
result.
For
So
the
the
example,
cut-off value can
as in
be
the detection
calculated
limit
by
is about
the
80
value
microgram per
the false
liter,
negative
and the
is
maximum
below
permitted level
5%,
is about
something like this.
125
So it is
microgram
possible.
per liter.
It is possible
So
that
by
the detection
doing experiment,
limit
it is nice
below the
10 times its
below
of
cut-off
concentration,
value, and the
and we
cut-off
can
value should
calculate
below
the
the
positive
maximum
result
permitted. I will summarize
2014.
the important
And
aspect
then
of
I
this
continue
application.
with biological assay. The bioactivity of extract or isolated compound can be determined
Bioassay
using some biological
Bioassay
assay.
can be defined as analysis
As chemical
of
assay,
drug
the method
to
also
quantify the
should be
biological
validated.
activity of one
This
or
is
more
the
component
complete
by determining its
our
capacity
recent
for
publication,
producing
just
an
only in
expected
press.
biological
I will
activity
summarize
of a
the
culture of
important
living
aspect
cell
of this
in vitro
publication.
or a test organism in vivo. That is the
Hello?
definition of the USP 43. Maybe
Bioassay can be defined as analysis of
but the aim of
drug
performing
to quantify the biological activity or activity of one or more component by determining its capacity for producing
or a biological test system. For doing that, this is some steps should be taken.
an expected
First, we
biological
should
activity
do
living
literature
cell
survey
in
for
vitro
selecting
on a test
correct
organism
bioassay.
in
I
vivo.
mean,
That is
what
the
we have,
definition of the USP
the equipment,
43.
what we have the
Maybe
cell, so we should survey from
other guardians have a
literature
different
and then select.
definition.
After
But the
that,
aim
we
of
should
performing
evaluating the
biological assay
stability
or
of the
bioassay
extract or
is to
compound
determine
of the
the
selected
effect of a
solvent,
substance
and
on
then
a certain
after
type
that, we
of
should
living
testing
matter
the
or a biological
performance of
test
the
system.
biosystem using
For doing
positive
that,
and negative
this
standards.
is some steps,
Also the same thing with
steps should be taken.
material, testing
So
material.
first, we should
then
do
determination
literature
of
survey
visi
for selecting the correct
determination of
bioassay. I
accepted
mean, what we have,
determination of
the equipment, what we have, the cells, so we should survey from
using
the literature and then select.
We can decide
After that, we should
how to study
evaluating the
using
stability
animal
of
study
the
or
extract
clinical
or
study.
compound of the selected solvent.
All this cell
And then
line should
after
be
that, we
first
should testing
authenticated
the performance of the biosystem using positive and negative standard. Also the same thing with material, testing material, and then determination of EC50, determination of selective index, and determination of accurate relative potency of sample accurately using authentic standard. And we can decide it how to study using animal study or clinical study.
Stable.
All these cell lines should be first authenticated although we have
This is a method. You can download this method. Selecting the organism, cell line, or bioassay should be based on the used formal guardians. For the example, WSO or other guardian. Leishmaniasis, we shoot this cell line. This is a number of ATCC. so on, you can have this guide from the WHO, etc. There should be certain timeline. The biostatic should be selected and sample, then calculating the Z factor. Is standard or sample. That is the equation. That is the maximum. That is the minimum. Bottom minimum. That is the calculation. For Z factor, that is a standard deviation of positive control, negative control. That is the average positive control, average data, and this is average negative of negative control. This is the equation, the same equation, but this is the requirement. So it means if we do something using certain cell line, what we first to do is using the suitable standard to determine the Z factor. The Z factor must be more than 0.5 or at least not negative. That is the requirement. If the Z factor is not acceptable, it means that the system is not suitable. After the system is suitable for the standard, the same thing we should do with our sample. The sample should also give acceptable Z factor value. This is very important because without this method, we cannot show whether our biosystem is okay or not, right? Our biosystem is okay or not for our purposes. So that's why we should try to evaluate this biosystem, I mean the cell, the standard, etc., using this equation. After that, we continue to determine what they call selective index. Selective index can be defined as the ratio of the toxic concentration of a sample against its effective bioactive concentration. The ideal drug should have a relative high toxic concentration, but with a very low active concentration. This is the equation. This is IC50 for cancer. This is a cancer cell, no cancer cell. This is for cancer cell. SI should be as high as possible. 10 is nice, but below 1 is not acceptable. If the value 1 to 10, we should try after experiment, after biosystem to confirm the result. If the selective index is too low, it means the compound or the extract is too toxic. I think it's no need to further investigate it because it is too toxic. For continuing the work for in vivo or clinical study, the accurate potency of the tested material must be determined. This is needed to decide the correct administration dose if we want to do animal evaluation or clinical evaluation. For this purpose, the author, I recommend you to apply the method of European Pharmacopoeia 10/2020 section 5.3. This section gives you complete equation. We can just set the equation into Excel and then we can do manually with complete with many parameter or many conditions. Or we can use also from USB 43, but the equation is not complete. I recommend using computer, but I recommend this using European Pharmacopoeia. That is nice. We can do it manually by Excel only. This is the conclusion and recommendation. Indonesia is very rich of traditional herbs and other natural resources that can be developed into phytopharmaceutical and or modern drug. It is recommended to determine the quality marker and its specification range, I mean the maximum and minimum content of each of the herbal monograph that's described in Pharmacopoeia Herbal. Performing clinical trial for all marketed phytopharmaceuticals is needed to have a consistent quality of every batch of the product. If we have consistent quality of the product, I think the medical doctor will very happy to apply this herbal drugs. For having reliable conclusion, all used chemical and biological assay must be first validated according to the newest guidance. before it can be applied. If the use method were not first validated, the conclusion could be wrong. Due to the limited funding and facility in Indonesia, collaboration between scientists are very important to develop the phytopharmaceutical or modern drug from Indonesian herbs. Coordination between university and research institutes are very crucial to prevent the duplication of research, and the limited funding can be efficiently used. I think if we can do collaboration from all university in Indonesia and LIPI to be the coordinate all the research, I think it'll be nice. This is, I will thank to Dr. Abdi for the nice invitation. And once again, thank you for your nice attention. It will be very nice if we can do collaboration. Do you agree, dear distinguished participants? Thank you. Good day. Nice to meet you all.
Okay, thank you very much, Professor Gunawan. Now we still have about 15 minutes before Professor Chaudhary join with us. So maybe we will have a little discussion. Yeah, audience already raised her hands. One audience, Ibu Emma Damayanti, but we don't know from which institution. She wants to deliver her question to Professor Gunawan directly. Maybe organizing committee can unmute Ibu Emma Damayanti. Halo, Ibu Damayanti. No active. Okay. Maybe we move to question delivered from Q&A facility. Yeah, one question come from Pak Ahmad Saripudin from LIPI. The question delivered to Professor Gunawan. The question is, for small enterprise that produce certain jamu, should they do the assay of their jamu by themselves? If the assay is conducted by government research institute or other, how about the patent, intellectual property right or property of the compound or jamu? Is it belong to the jamu producer or the assayer? Okay, please, Professor Gunawan, can you explain and answer about this question?
What I'm talking about is how to do a complete validation method or how to do quality control. This will be done if we have equipment. We need many equipment to do that. But if it's not, I think we can do very simple. We can do by the public jamu or the home industry can make a note at first with very simple to note all of things regarding the drugs, where they got the drug, where the place, and the age, the part of the plants, and then to note also how to make the drug, and then they can ask the consumer, how about the drug? Is this okay or not? By doing this, someday, if we have complete data, we can conclude if the plant is collected in a certain places, they will not active. Or if the plant collected with certain is, that is not active. That is very simple, but we need to do some survey and we do a work There are many people, and we can collect together all the data, and we can do something metabolic profiling, but without any chemical method, without any, but just be asking the people, the consumer, about the drug that they can produce, because the drug may be produced with different way. This is my question. This is nice if someone do this survey, all of the survey, and then at last we have conclusion. This plant is active if cultivated on a certain place. This plant is active if harvest on certain months or something like that. So the plant is active is the mode of extraction, something like this. At last we will have something like that. But if we have chemical equipment, that's say we will do profiling, chemical profiling, we will do IOC profiling. Regarding the patent, maybe the participant know better than me. I do not know about regarding who will get the patent of this research. I think both of them who work with this work will get the patent. That's my answer. Maybe it's okay.
Okay. Thank you, Prof. Okay, thank you, Prof. Gunawan. Maybe before conduct the research, if jamu producer and researcher would like to have joint research, maybe better to communicate in the before conduct the research about the property right, maybe. I see. Okay, we still have time. We will move to second question from Ibu Marisa Angelina from LIPI. Question also to Professor Gunawan. It is a nice presentation and also full of knowledge, Professor. How do you think in Indonesia is the regulation body like BPOM applied the rule of marker of herbal medicine in standardized traditional drugs or phytopharmaca? Okay, please.
Okay, thank you for the nice question. As I described previously, Pharmacopoeia Herbal Indonesian or USP 43 or European Pharmacopoeia, they just only describe the minimum standard, the minimum concentration of certain compound or a group of compound. This is generally something like that. The maximum compound, the maximum concentration was not described. This is that's why by this reason Professor Ringer wrote in his publication that is not enough for ensuring the quality of the drug, because if it is just determining the polyphenol or flavonoid or certain compound, it would be not correlated with the activity. The drug will be okay if they can be used by the customer. So that's why it is need to determine the quality marker or marker ranking system 3 to 5, which from the plan the actually quality marker. Unfortunately, in Pharmacopoeia Herbal Indonesia and other recent Pharmacopoeia USA, this was not described. This is only described by some publication, but just only a few. That's why if we do quality control using any marker, maybe it is okay, but the pharmacology action is not the same test to test. This is the problem. That's why it is nice if we start from Indonesian medicinal plant one by one to search what is actually the quality marker. This is not yet described in any pharmacopoeia of the world, current pharmacopoeia.
Okay, thank you, Professor Gunawan. Maybe
if Ibu Maris have another comment of my answer, let me know.
Yes, Professor Gunawan, I can accept your idea. Yes, I think like that in Indonesia, the BPOM cannot apply all of the rule of market that you say in your presentation.
So that's why it's nice if LIPI or BPOM coordinate the research in all Indonesia's one by one. So there is application, there is no efficient. We can think that's nice. That's my dream.
Yes.
10, 40 years ago.
Yeah. Nice, Professor Gunawan. Nice. Thank you very much, Professor Gunawan.
Okay, thank you, Ibu Marisa. Yeah, maybe we move to another question. Now, question for Dr. Ambara from Pak Ahmad Sarifuddin from LIPI. The question is, as we know that the cure treatment for cancer is very painful, are there any non-invasive or destructive method development by Japan to cure cancer? Okay, please, Dr. Ambara, can you explain for us?
As far as I know, in Japan and also in several other countries, I guess, for the treatment of cancer, actually there is no general way to treat all cancer. I think every type of cancers, even the same cancer in different person, also need to be treated in a specific way. So also the method, there are always the advantage and the disadvantage. So in my case, I focus on the first part of my talk. I focus on the method to support the surgeon during the diagnosis during the cancer surgery. And on the second part, we use this product to eliminate the off-target reactions. So actually, it depends on the stage whether we can do the non-invasive. But actually, even though we did perform the non-invasive, it doesn't guarantee that in the future the cancer will not regenerate again. So, as far I think so far also in the advanced country also, there's the way the method is still very, could be different for each person. Like that's even in Japan as far as I know.
Okay, maybe it's clear. There is more question to Dr. Ambara from Ibu Sylvia Lekatompesi. Yeah, the question is, my cousins, young person use traditional medicine term. Okay, can you explain, Dr. Ambara, please?
When we start to start our research, our purpose is that we would like to make a contribution for the cancer treatment. So at the first part of my talk, I mentioned about the chemical probes that has been used in the hospital in Japan. It's easy to be approved. The method is easy to be approved because on that method, the cancer was taken out from the patient bodies. So it was taken outside from the cancer, from the patient bodies, and then we treat the stump by our probe. So we did not put any reagent into the human. So that's why it's easy to be approved, and all it's already start in the Osaka University Hospital. But on the second part, on the chemical, on the prodrug that I have been talk on the second part, it needs to be injected into the patients in the future if it's approved. So there's still a lot of several steps to be tested, but on the animal level, it shows a very promising result. So we are trying hard to make it fast to be applied further in the near future. So hopefully we could contribute in this area also. Okay.
Okay. Ibu Sylvia also asked her cousin use traditional medicine, turmeric and suchile. Do you have comment for this part?
So actually, I'm not the expert for this traditional medicine. Professor Gunawan maybe can answer this question, but we are focusing on the drug, on the pure drug that can be used can be linked to our product. So yeah, I cannot comment on these traditional medicines.
Okay, it's okay. Okay. Already 10:30. Ibu Marisa, Professor Chudari is already joined with us or not yet?
I think not. I think not
Pak
yet.
Abdi, maybe no.
Maybe we can extend for discussion around 15 minutes more.
Okay.
Because Prof. Iqbal has not joined us.
Okay. Okay, let we continue to discussion. Next question come from Carla from USN Kolaka. The question delivered to Dr. Ambara. Is the probe could be used to detect a very early cancer stage? Okay, please, Dr. Ambara.
So our collaborators in Osaka University Hospital, the breast cancer surgeon, when we published the paper, he tested on several stages of cancer, even stage 1 to stage 4 of cancer, and our probe could be accurately detect the cancer for this kind of breast cancer. So that could be used to guide the surgeon until which area need to be removed, so that the reoccurrence can be we can just the recurrence hopefully will not occur again in the future. So yeah, we tried with from the phase 1 to phase 4 of the breast cancer, and now also more data has been collected, but for the details I could not say more about the result for this point. But on our previous publications, we tested on the phase 1 to phase 4 of the cancer patients.
Okay. It means it's possible to use in early stage.
Yeah, hopefully, yes.
Yeah. Okay, maybe it's already clear. Okay, we move to next question. Come from Ibu Teni Ernawati from LIPI also. The question is, it is nice presentation and also full of knowledge, Dr. Ambara. From the results of the research you have done, are there always significant differences for cancer cells and normal cells against the anti-cancer chemical compounds that have been synthesized?
Okay. So first of all, I think this is Buteni from which come from to Osaka several years ago, if I'm not mistaken.
Yeah, probably yes.
So yeah, I mean, we tested to several types of normal and cancer cells with our products, and they give almost the similar results. So we could see the difference between the normals and the cancer cells. So far, I show you only one drugs in this experiment and these presentations, but actually we tried several more drugs, which could not be shown now today because we still need to publish and patent the data. And everything seems good. They give the clear difference between the normal cells and the cancer cells. I'm sorry I could not show the data for now, but hopefully on the different opportunities I could show you in more details after we publish the results in the near futures. Thank you.
Okay. It's already answered the question. Maybe we move to another question. Okay. Yeah, next question come from Ibu Sofa Fajria. The question is, thank you very much for nice presentation for both presenter. I would like to Professor Gunawan about the quality marker, how we can get the reliable data of the marker. Is it possible if we only examine only one tool, such as TLC scanner or densitometer? Thank you. Please, Professor Gunawan. Okay, sorry, your voice is still mute.
Okay. Sorry. Thank you for the nice question. I think for determining the quality marker, we should do some chemical profiling and some ISAC profiling. For chemical profiling, we should have LC-MS. That's impossible if we have impossible if we just only using TLC. If we have TLC, and then we should isolate the fraction of the fraction of the spot and determine the structure. That is my answer. But if we have LC-MS, we can directly predict or determine the quality marker. If only using TLC, we can do the same thing, TLC profiling and ISAC profiling, and by combine with using multivariate, we can predict which spot that active, which spot that is not active. The active spot can be isolated and the structure can be determined. That's my comment. Maybe.
Okay, thank you, Prof, for the answer. It's very difficult for us. Thank you very much.
Okay, thank you. I already saw Professor Chaudhary already joined with us. Welcome, Professor Chaudhary. Maybe your voice is still mute.
Assalamualaikum warahmatullah. Good morning.
Waalaikumsalam warahmatullahi wabarakatuh. Welcome to our symposium. Okay. You are ready to present your presentation?
Yes, inshallah, whenever you want me to.
Okay. I will introduce our third keynote speaker, Professor Iqbal Chaudhary. Professor Chaudhary is a distinguished national meritorious director at the International Center for Chemical and Biological Science, HEJ Research Institute for Chemistry, and Dr. Panjwani Center for Molecular Medicine and Drug Research, and Coordinator General COMSTEX, Organization of Islamic Cooperation Standing Committee on Scientific and Technological Cooperation. He has written and edited 76 books, most of which have been published in US and Europe. He is also the author of the over 1,000 research paper. Wow. In top international science journal of the West, as well as 55 US patents. He has been among the most cited scientists in Pakistan in last 5 years, with citation exceeding 25,000 citation, with H index 68. Professor Chaudhary has skillfully used his deep understanding of chemical principle and biological process in the discovery of a large number of fascinating molecules with a potential therapeutic application. In this morning, Professor Chaudhary will present the presentation titled Integrated Natural and Synthetic Chemistry-Based Approach in Heat to Lead in Drug Discovery, example of our recent work. Okay, please, Professor Chaudhary, time is yours.
Bismillahirrahmanirrahim. Assalamu alaikum and good morning, ladies and gentlemen. Thank you very much, Professor Andrea Augusta, for a very generous introduction. It's always a pleasure to speak to our brothers and sisters in Indonesia. Indonesia is my second home, and it's always a pleasure to be with you. So I'm absolutely delighted to be part of this interesting conference, though I really wanted to visit you physically, but hopefully, inshallah, soon when situation will improve, we would be able to have this event physically. So what I would do is to share with you my slides. Let me take you through slideshow, please. Okay, so next 45 minutes, I would be speaking to you. About this interesting work which we have been carrying out since last several years, and that is the use of natural product and synthetic chemicals in the discovery of medicine. And as you have mentioned during my introduction, that we have got many patents from different countries, and we have developed many lead molecules which are in different phases of development. I'll just give you example of a few. It's very important. You work in LIPI also. Many places in the developing world, we have a huge reservoir of natural products, and we have been using them for a variety of different purposes. And these are examples of some natural products which have made to the world scene as drugs. So whether it is quinine or morphine or Taxol, all of them are extremely important, and they have been discovered out of ethnobotanical practices and the science mixed with it. In Pakistan, we have a long country, not as big as yours, but this is a one single piece of land, about 2,700 kilometers long from this corner to this corner, an extremely diverse country, because from tropical shores to deserts and subtropical areas to Siberian climate, all mountains and lots of snow. We have very diverse flora, and that diverse flora allow us to work on very large number of natural products. And in the process, we have collected over 5,000 plants. We collected those plants along with the information of their use in different cultures for therapeutic purposes. And then out of them, we have conducted lots of different studies. So primarily information about their folk use, and then by using scientific methods, bioassays and phytochemistry, we were able to isolate very large number of compounds. I'll just give you example of a few which made it to the US patent, and they are in different phases of development. Isox, which was isolated. Isox is the name of a drug candidate which is in phase 2 clinical trial. This was isolated from Delphinium dentatum, and it is one of the strongest anti-epileptic natural products. Then there is a health supplement which we have developed for Parkinson's disease. This is also recently patented in USA. We have also developed topical formulation for various skin diseases, including leishmaniasis and that is also patented. I'm going to present to you briefly about three projects, and these three projects based on very large number of natural products and synthetic compound which we have in our library. So we have a large bank of diverse natural and synthetic compounds, and these compounds have very interesting structures, and these structures are primarily the basis of their function. But before that, I'll just show you what new skeleton we have introduced into the world literature. World literature is now full of our discoveries. You would find from a one single group, as much as we have published, no other research group in the world has introduced so many chemical compounds in the field of natural product chemistry. For instance, this is a triterpene, which we have isolated in fascinating cage-like structure, which we have isolated from a plant, and synthesis of this compound was the major challenge. This is an interesting molecule which we have isolated from Nigella sativa, which is a very famous herb, which is a very famous spice, and used by our prophet. And this is also a very interesting molecule. As you can see that this is a skeleton in which we have a positive charge, is quinazolidine skeleton. And this is also another very interesting compound with N oxide isolated from Nigella sativa. This is a diterpene which was isolated from a marine from marine source. As you can see that there are lots of interesting skeleton because these are secondary metabolites, and gives a fascinating structures which may have very interesting biological activities. Zoanthamine, a very interesting steroidal molecule, a series of compound which was isolated from a marine plant, and of course lots of other interesting molecules, but just wanted to give you the glimpses of absolutely interesting structures which you can expect from natural products, because nature provides you molecular inspiration that gives you interesting skeleton which you can overbuild. Now, this is the first project which I wanted to discuss with you briefly, and that is related to resistance in microorganisms. We all understand that bacteria, fungi, and parasites are developing resistance, and these are bad bugs. You know, for instance, it's called ESCAPE, in which we have 6 of these microorganisms declared as some of the most dangerous microorganisms in the world, and they have developed literally resistance against all known antibiotics. I'll give you example of our work, though this is a big project and I can speak about that for several hours, but I'll just tell you how important it is to work in this particular field. Resistance is everywhere, and we all are affected from microbial resistance. When we get sick, it becomes very difficult for physicians to treat us because microorganisms are often resistant against the available antibiotics. Now, resistance is a cross-cutting, is everywhere, from viruses to bacteria to parasites, and you know there are lots of different reasons, but this is a globally alarming situation. Now, there are 3 different options in which one can work. You can discover new antibiotics. That takes lots of money, about $2 billion, about 10 years, and you never know how long this antibiotic would continue to function because resistance is often emerging within a decade. The second option is structural modification of existing antibacterial agents. This is also an interesting approach, that you take existing antibiotics, you modify their structures, and that you hope that this would work for a while. And that is also a good approach. And many of the antibiotics which you see in the marketplace are the modified form of existing antibacterial, antimicrobial agents. But what we have done in our institute, in our research group, is to actually use agents which can modify and modulate resistance in antimicrobial in microorganism. Primarily, the objective is that you have antibiotic, and then you have a modulator which help the antibiotics to work. So these are called resistance modifier. These are so-called resistance, which is epidemic methicillin resistant Staphylococcus aureus, 17 species. And you can see that this is active more than the vancomycin. So this is a molecule which was found to be more active than the drug of choice, vancomycin. This is another molecule which was isolated from the same plant, and the only difference between the previous one and this one is the presence of this isopropyl unit, but the activity has reduced. I'll just take you what we have done. So we have selected this one compound, and we have carried out lots of morphological studies by using microscopic methods, and we have done lots of molecular studies by using flow cytometry. And by combination of these methods, we were able to have some idea of the activity of this molecule, which is called arginine. This is Staphylococcus aureus, which we have targeted. We all understand Staph aureus is an extremely important bug, is gram-positive. It is nosocomial and is also community acquired, is a normal flora of human body. And but that become pathogenic because a lot of systemic and lots of localized infections are caused by Staphylococcus aureus. This is a picture of a normal colony of Staphylococcus aureus, and when you expose this colony with this particular compound, arginine, then you see a very interesting structure. You see that the colony is simplified, not many bacteria survived, and even the one which survived, they have different morphology. So morphological changes were noticed through microscopy, and by understanding, for understanding the effect of it, we have carried out a very interesting study in which we have used propedium iodide and compound and antibiotic. And as you can see that without the compound, without this compound, the number of cells which accept propedium iodide are very few. But when it is in combination of compound and antibiotic, then you can see this red region, which means that larger number of microorganisms have accepted the dye, and which actually indicate that this compound is capable of helping antibiotic to enter into the cells. And this is scanning electron microscope, in which you can see the grape-like structure of Staphylococcus aureus, AMRSA 17. But when it is exposed to this particular compound, a completely different behavior is seen. Morphology changes. You can see the cell division is abnormal, biofilm is broken, and at the same time, you can see that the colonies are simplified and cells are disintegrated. This is an interesting picture, transmission electron microscope, in which you can see these cells nicely dividing in two parts, binary fission in a normal colony. But when it is exposed to arginine A, then the morphological changes are so obvious that you can see the cells are abnormally dividing, cells become hollow, the subcellular organelle come out. But this is a fascinating one technique in which you can look into the cell at the nanoscale. So you can measure the cell height, you can measure the cell dimension, and the color represent the cell height. So on the scale side, you can see by using atomic force microscopy, the cell can be measured at a nanoscale. This is the normal cell. And when this single cell, which is about 27.5 nanometer in height, when it is exposed to arginine, then amazing thing happened. You can see the cell height has collapsed, cell collapsed, to 0.75, literally nothing, and you can see most of the organelles in the cells of Staphylococcus aureus come out of it. So complete disintegration of cell is measured, and is an extremely interesting technique in which you can see morphology and measure what happened to the cell in real time, because you don't need to dye them, you don't need to fix them, you can do it in real time. So you can take the picture of cell, and you can look into the behavior of cell in the presence of this compound. Now, why it actually happened is still a mystery. We are still working on it, but one theory on which we have worked and found some indication is that this compound arginine is capable of inducing the production of reactive oxygen species, peroxide. And so this bacterial cell, in the presence of arginine A, produced lots of reactive oxygen species. And as we know, reactive oxygen species are free radicals. They can damage the cell wall, they can damage lipid membrane, and they can also damage DNA. So a whole range of damage was actually observed in this case, and this may be the mechanism of cell destruction. There's some antibiotics which function through the induction of reactive oxygen species. This is an interesting slide in which you can see on the side various antibiotics. So you can see erythromycin, streptomycin, clindamycin, so different antibiotics. And these four bars represent what actually happened. In the absence of, I'll give you an example of clindamycin. In the absence of this molecule, you need to have a large quantity of clindamycin, about 2,000 microgram per ml. But in the presence of this arginine, you need only 8 microgram per ml of clindamycin. So this molecule is capable of helping classical antibiotics to enter into the cell and function. Now we have worked on understanding the chemistry of cell, and this is a protein which was isolated from Staphylococcus aureus resistant strain. So we know that there are certain proteins which are different in susceptible strain and resistant strain, and this protein was expressed, its structure was done by using NMR spectroscopy. And here you can see the structure of a protein which is different between susceptible strain and receptor strain. Similarly, a number of other proteins were isolated, and this study was conducted by using various spectroscopic techniques. There are some of the international patents which we have obtained on resistance reversal agents, not an exclusive list of patents. There are many more. But I'll just, because I still have about 20 minutes, and I just want to take you to 2 more projects. And this is related to, again, a very interesting work which we have conducted in last 6 years or 7 years. We understand diabetes is a disease which is affecting lots of people. There is no medicine available for diabetes, and the ultimate aim of anti-diabetic therapy is to produce insulin in enough quantities in our body that is capable of handling the sugar, which is called postprandial sugar level. So achieving normal glucose level in blood is the outcome of it. And there are various types of drugs which are available to handle that. Now, what actually happened in diabetes is something very interesting. You know, we keep using anti-diabetic medicine, but we slowly and gradually start losing pancreatic cells. Now, pancreatic beta cells are extremely important because they are the ones which produce insulin. And slowly and gradually with chronic diabetes, the number of cells start decreasing. And as number of cells start decreasing, capacity to induce insulin within the body become hampered, and our body cannot really produce enough quantities of insulin. So Beta cell mass and beta cell function is extremely important. And why it is important to protect beta cell mass? Because we need to have insulin produced in enough quantities in response of glucose. There are many drugs available, but of course, ultimately, the actual strategy is to prevent beta cells from apoptosis. Why beta cell die? Because diabetes is unfortunately a pro-inflammatory disease. This is a disease with a chronic inflammation, and in diabetes, there are lots of cytokines which are produced in our bodies, which eventually kill beta cell mass. And that is the model. What we have done was that we have used 3 pro-inflammatory cytokines, and we use beta cell. And by using these pro-inflammatory cytokines, we induce apoptosis, programmed cell death in beta cell. And we wanted to mimic what actually happens in a chronic diabetic condition in a patient. So what actually happens is that many of these cytokines start attacking beta cells and their number decrease through apoptosis. We wanted to develop a very good model. So we have this cell line, which is beta cell line, and we had these 3 cytokines, and we use this cocktail of cytokines in order to induce apoptosis in beta cell. This is the model which we have used. We have screened about 1,500 compounds of natural and synthetic origin. And and what we have done was that we looked into what actually happened in the presence and absence of these compounds. Now, in the absence of these compounds, beta cell die, because these cytokines are capable of inducing the cell death. And this can be measured by caspase-3 activity, by measuring ATP level. ATP level is the indication of cell viability. If low ATP level is detected, it means cells are less. Caspase-3 is an enzyme which is responsible of apoptosis. So if there is a high level of apoptosis, you see high level of caspase-3. So there are various molecular markers through which you can see whether your compound is capable of protecting beta cell from apoptosis. Lots of different natural products were screened. I'll give you example of only one, arginine, which is a very famous natural product. And this has been isolated from a large number of plants. This is a dihydroisocomarine loaded with lots of hydroxyprope isolated from various medicinal plants. Arginine is a very common natural product, and you can get them in any quantity because this is a very common secondary metabolite which can be isolated from various plants. What we have done was that we used this cell model, beta cell of mouse, and then we induce apoptosis by using cytokines, inflammatory markers, and we use arginine. So here you can see that in the absence of arginine, you see this is the normal cell without cytokine. So you can see that ATP level is high. In the presence of cytokine, the ATP level is very low. It means few cells. And in the presence of compound, arginine, the number of cells in a dose-dependent manner, cells are protected, and ATP level is as high as physiological normal cell condition. Would demonstrate that caspase-3 is very low in normal cells. It's very high in cells which are insulted with cytokines. And in a dose-dependent manner, this compound is capable of reducing the caspase-3 in a dose-dependent manner, which means that it is inhibiting the apoptotic process. So this is the kind of studies which were conducted. This is mitochondrial membrane potential. Diabetes is a mitochondrial disease in which mitochondria get sick, and mitochondria has a membrane. and this membrane potential is an indication of the health of mitochondrial function. So this compound was capable of restoring the mitochondrial membrane potential, which is the indication of. But ultimately, I'll just show you what actually happens. What you want to see is primarily that when glucose is provided to beta cell, the cell should be able to produce insulin. And this is an interesting slide because here you can see that in a dose-dependent manner, dose of a glucose, this compound was able to protect the cell, and as a result of it, the insulin are produced in large quantities. So this is insulin secretion, and that indicate that this particular compound is capable of not only protecting the beta cell mass, but also the function. Function of beta cell is to produce insulin. And here you can see insulin produced in response of glucose, which is a gold test. This is a very interesting topic, still not fully explored. And a lot of studies are being conducted now in the world because this is a new line in which one can develop anti-diabetic medicine. The last project which I will present to you is for the young people. Biotransformation has always been my favorite because we often do lots of synthetic chemistry by using chemicals, catalysts, solvents, and different kinds. Why don't we use microorganisms? Why don't we use bacteria? Why don't we use fungi? And why don't we just carry out these fascinating chemical changes by using cells? So in our laboratories in Karachi, we have shelves full of bacterial cells, we have fungal cells, we have plant cells and animal cells, and we use these cells as our chemical agent, so-called chemical agent, because it's fascinating. You don't need to use chemicals. The only thing which you need to use is the culture of these cells. And this process is called biotransformation, a very fascinating branch of chemical derivatization because it provides you very interesting molecular analog. So science is extremely simple. Methodology is very simple. You have, of course, cultures of different kinds, and you have your compound, and you inoculate your cultures with the cells, and then you keep them for incubation for a certain period of time. Once the cells, the bacterial cells or fungal cells incubate the compound, you would be able to observe the changes, but for that you need to extract out your compounds by using simple extraction method, purification. You characterize and you carry out biopsy screening. So primarily the objective of this integrated approach is to use living cells instead of using chemicals. And the advantage of this field is that you get a fascinating new derivatization. Chemical derivatization are very predictable, that only occurs at the active site, but when you use biocatalysts like cells, then the structures of molecules are very interestingly modified, and you get often novel analogs. This is an example of the use of biotransformation, and that we have used for the discovery of aromatase inhibitors. Aromatase is a very important enzyme in the treatment of breast cancer. Breast cancer, as we know, is unfortunately very common disease throughout the world. It is the second major cause of cancer-related death in women. And this is because of the abnormal growth of cells at different regions of the human breast. And most of these breast cancers, 2/3 of them, are because of estrogen. Estrogen is an extremely important growth regulator. It's a hormone which is produced in very large quantities in human ovary, in breast and peripheral tissue treatment of breast cancer after surgical removal and after making sure that the recurrence of breast cancer do not really occur again. So treatment option is that you inhibit the enzyme which is responsible of the synthesis of estrogen. This is an interesting slide because here you can see that these are testosterones which are converted into estrogen. Estradiol is in estrogen. This is androstenedione which is converted into estrone. They both are estrogen, and these estrogens are required for the cell division. Without them, cell would not be divided, and you don't want cells to be divided. So therefore, you need to inhibit the synthesis of these two compounds, and that can only be happen by inhibiting the enzyme which is responsible of the synthesis of estrogen. So aromatase is an enzyme which catalyzes the synthesis of estrogen. This is the last step of estrogen synthesis, and in order to inhibit aromatase, you need to develop aromatase inhibitors. And when you have aromatase inhibitors, which are the class of drug, very important class of drug now, then estrogen would not be produced and cell would not be divided. And this is a way of making sure that cancer do not occur or cancer progression is controlled. This is the structure of estrogen. Aromatase enzyme is a complex enzyme, very large molecule. And these are some of the aromatase inhibitors which are available in the marketplace. Our approach was that we wanted to synthesize aromatase inhibitors by biotransformation. And for that, we use various different kinds of steroids because steroids are close in structure of estrogen. And we have then developed a new analog. So the first problem was to where to get the aromatase enzyme. Aromatase enzyme is not easily available. You cannot really order it. It's a very difficult and extremely expensive enzyme to purchase. Some people would produce that for you, but they would charge literally thousands of dollars. And we found a very easy way, human placenta, plenty of it, produce aromatase enzyme, and microsomal cells of placenta contain aromatase enzyme in very large quantities. But we have also, by using recombinant technologies, we also have expressed aromatase in the cell, E. coli. So we have a very pure form of aromatase enzyme and also aromatase enzyme from human placenta. Now, how to measure the activity of aromatase is just to measure the production of estradiol, which is an estrogen, by using ultra pressure liquid chromatography. And I'll just give you one example. I don't want to take much of your time. I just wanted to let you know that this is a synthetic steroid. And here you can see the wonderful chemistry, what happened in biotransformation. So here you can see that this is test electron. And by using two different fungi, Fusarium linea, you can, and the other is amphisalina, you can modify the structure. So you can see that in the structure, there is one oxygen ring, one hydroxy group is introduced, different stereochemistry. And this is the standard drug, exmestine, with which we have compared the structure. So here you can see very well that we were able to produce new libraries of aromatase inhibitors. You can see IC50 value 0.82, which is not different from the standard drug available, but some of the molecules which we have produced were found to be even more active. This is 0.33, and as you can see that this is also as active as the standard drug. The message which I want you to convey here is the following. You can create new chemical space by using biotransformation. Very simple method, and by doing that, you often produce novel analogs. So here you can see many structures which are as active as the standard drug. I don't want to go further on it. This is, for example, this particular molecule is several-fold more active than the standard drug. So we have introduced new analogs of aromatase inhibitors. And these aromatase inhibitors are much more active than what you can see in the pharmacy shelf. And that we were able to do only by using a novel approach. And of course, having a science at the interface of biology and chemistry. So we do lots of interesting synthetic chemistry. We do lots of natural chemistry. We do lots of biotransformation, but as you can see, the major strength of our work is that we have a strong biological background and that we have very experienced scientists working in the field of various fields of biological sciences, from molecular biology to structural biology, from cell physiology to molecular microbiology, whole range of it. So what I wanted to present to you, which is briefly summarized, that it is possible to use natural products and to develop new lead molecule as drugs. And it's also possible to take whole molecules and transform their structure by using different combinations of biotransformation. Let me introduce my research group. I have very talented young colleagues, and they are expert in their own fields. By own merit, they know science at a very high level, and they work in combination, in tandem together with me. And we have been very fortunate to have funding from lots of pharmaceutical companies. This is the institute we work. This is famous ITJ Research Institute of Chemistry, which was established back in 1966. Is one of the most famous natural product chemistry institutions in the world. This is a molecular medicine center. This is Dr. Taruman Laboratory, who is our patron-in-chief and a very famous scientist in the field of bioorganic chemistry. This is biotechnology, where we do lots of medicinal plant cultivation. This is a nanotechnology center. We have big genome centers. We do lots of genomic work, forensic DNA, and this is a center which we established in recognition of the services of a German scientist. We have our own laboratories at the shoreline of Arabian Sea and also on the mountains. This is our National Institute of Virology, which is a big center doing lots of virus-based research. These are animal houses of different kinds, and of course, lots of. So this is our clinical research unit. We do lots of clinical studies on human patients by using our own hospital. We have a 120-bed hospital. We are right now in the process of conducting phase 1 and phase 3 clinical trial of COVID-19 vaccine. This is our industrial and medical center, our technology incubation centers. And of course, our facilities are among the finest in the world. We have 13 superconducting NMR machines, lots of X-rays and mass spectrometers of different kinds. But I think the most important thing which I wanted to mention here is the quality of people. So there are 17 research buildings, and each building is designed to do different kinds of research. We have our own guest houses and hotel on campus, and we get lots of visitors from all over the world. So we get 200 visitors every year. Indonesian students in the past also. We would look forward to have more of them coming to our institute. And our relationship with Indonesia is absolutely phenomenal, brotherly, and we are very close to each other. So thank you very much. Visit Pakistan. It's a beautiful country with lots of culture, lots of diversity, lots of history, and good food, very close to Indonesian food. Thank you.
Okay, thank you very much, Professor Chaudhary, for very nice presentation. Yeah. Now we have discussion time until 11:55. Okay. If you have some question, please write in Q&A facility. Yeah, before Professor Chaudhary joins, we already have a little discussion, so we will extend in this session. Maybe I would read the one question already appears in the chat room. The question from Mr. Ahmad Fatoni from LIPI. The question will deliver to Professor Gunawan. Thank you for sharing knowledge and lots of information related to quality control of herbal medicine. What are the strategies to develop herbal medicine in Indonesia due to the abundance of our biological resource? Whether herbal medicine research is focused on plants that have been used as traditionally or have never been used previously. And also, Mr. Fatoni delivered a question to Dr. Pradipta. Does the endogenous acrolein only occurs in breast cancer cells, or it can also occurs in other cancer cells, so that it can detect and treat for those cancer cells? Okay, please, Professor Gunawan and Dr. Pradipta answer the question. Maybe next question will deliver to Professor Chaudhary. Maybe we hope so. Okay, please.
Thank you for the question. I will try to answer. As I previously talked, the strategy is nice if we in Indonesia do collaboration between university or between research institute, so we can do we have limited facility, not as good as Professor Chaudhary showed. We have in our laboratory Indonesia very limited, so that's why we need to collaborate. But Balepom or IKPI or LIPI should coordinate the research. So the research should be coordinated, so the duplication or the same research can be avoided. So what shall we do is depend on the top. I think Balepom or IKPI should decide what plant should be first. What plant of, I mean what traditional plant, what we should do first. So by this, the Balepom or IKPI can ask certain university to do part of the research, and then we can combine. So by this method, the research will be fast and more efficient. As we know, at present time, many universities do research. I mean, almost university in Indonesia do research on herbal drugs. But I think most of them do the same thing, do the same topic. This is not nice. I think the most important is someone from the Jakarta should coordinate all the research in Indonesia, so we can have a good result in efficient and limited budget. I think this is the strategy that I recommend in my conclusion, and I have I always recommend this in my previous presentation and many seminar. We must do collaboration, but one or Balepom or IKPI or LIPI should be coordinator. Without that, that's very difficult. Thank you.
Okay, thank you very much for the answers, Professor Gunawan. Unfortunately, Dr. Pradipta already off from this room. Maybe the question will be delivered by email later. Okay, this is one question for Professor Iqbal from Dr. Arthur Lelono from Indonesian Institute of Science. The question is, Assalamualaikum, Prof. Iqbal. Thank you for your presentation. As we know that discovery of natural product took a long time, what is your opinion of using biotransformation? Will it slice the time needed in the discovery of natural products? And can we use deep learning in the structure activity relationships in the development of potent natural product for drugs? Please, Dr. Sheldon, Professor.
Brother Arthur, Assalamu alaikum. This is a very important question. I would say that these are two different approaches. And of course, natural product remain very, very important. But we also know that it is, you're right, that it takes lots of time and lots of efforts to isolate and to get enough quantities of a natural product. But of course, as you know, that lots of new technologies have been developed recently. There are two levels of technologies. Of course, we can do de-replication. We can quickly identify known natural products and new natural products by using hyphenated technique like LCMS, GCMS, LC-MS, NMR. And also, recently, there are lots of metabolic engineering is conducted now. It's possible to produce natural products in bacterial cells by identifying genes and then incorporating genes. By using recombinant technologies, one can produce various natural products. There is one approach called cell suspension culture. It's also possible that we have cells of the plant and we grow them in bioreactors, we produce them in large quantities. But you're right that isolating natural products is not trivial. It takes time. Biotransformation is extremely fascinating. It gives you novel analogs and is a very simple science, to be honest. You know, I really would like to encourage people to work in this field because of three reasons. Number one, that you don't need to have very expensive equipment. This is primarily a science in which you need incubator, perhaps some level of, you need to have extraction, you need to have incubations, and you need to have fermenters. You don't need to, even fermentation can be done on simple shaking bath. And second most important reason is that the structure of biotransformed products are not so different from the substrate. So identification of the structure is much easier than isolating a natural product which is completely novel and you don't have no idea about the structure. Often the biotransformed products are analogs of substrate, so their structure is not very difficult to elucidate. The third major reason is you can scale it up any level. You can produce them into any quantities by just scaling it up. You know, I mean, you do biotransformation on 1 liter, you can do it 100 liters also, and you can do it on 1,000 liter. It's sustainable science because the microorganisms you can produce as much as you wish. You can recycle the unconsumed substrate back to the loop. So because of these three reasons, biotransformation is still very exciting. But kindly remember that biotransformation would never give you novel, exciting, completely unknown compound. For that, you always have to look towards nature. Even synthetic chemistry would never give you that kind of structural diversity which you see in nature.
Okay, maybe clear, Dr. Arthur? Okay. Yeah, the biotransformation process is very, very interesting. We also conduct in our laboratory, Professor Chaudhary. Yeah. Okay, we move to next question from Ibu Marisa from LIPI to Professor Chaudhary. The question is, it's a nice presentation. Could you share about the results of your institution research related with the implementation? It have been done communication with some pharmaceutical industry, because sometimes in Indonesia, we have difficulties when we want to commercialize our products. Maybe you can suggest or something.
Yeah, Professor Marisa is also a very important problem which you have pointed out, an extremely important bottleneck which we all face. Now, there are two approaches. One is that if you wish to develop a pure natural product as ethical pharmaceutical, then you have to go through all clinical trials and everything. And that requires lots of money and lots of efforts, lots of time. We have been doing it in case of one anti-epileptic drug, which is called ISAC. This is in phase 2 clinical trial. This is the 11th year of development. It took about 11 years for us to reach to, and for that we got funding from a Canadian company, and that is a pure natural product, a natural product which we have then synthesized because you cannot get them enough quantities from the plant. So this is one approach. The second approach is, which I strongly believe is the one which we should really do, is for curiosity, you can always go and isolate compounds, study the mechanism of action, but always remember that if you would like to develop a pure natural product, then the cost is extremely high, because purification of a natural product in required pharmaceutical grade is extremely expensive. So I would always recommend that we work on composite medicine. We work on fractions, standard fractions. We prove their efficacy and we prove their safety. And that is a drug by all definitions. So for us in developing world, the best way of moving forward is to develop standardized natural products with safety, efficacy, and reproducibility with the required clinical data. And that is the best shot for us. An example which we have done is that we have recently, this is about $2 million project, which we are carrying out for a cell pharmaceutical company, a multinational company, for the development of nutraceutical for Parkinson patients. Parkinson patients are often very symptomatic, and there is no medicine available which can provide them a complete relief from symptomatic issues. So this dietary supplement, which is now internationally patented, and I would be happy to deliver a lecture about that separately, offline, is developed in collaboration with Searle. But again, this is a combination. This is not a pure natural product. This is a combination of drug and a combination of dietary substances, plants, and by using different animal models, we were able to develop that Parkinson nutraceutical. Nutraceutical is an area in which we must move in. That is an extremely important area which is easy to track and relatively easy to get into.
Okay. It's clear, Ibu Marisa?
Yes, very clear. Thank you, Professor Chaudhary. Thank you, Prof. Andrea.
Okay, thank you very much. Okay, we move to next question from Dr. Abdi Wira. The question is, thank you for a nice lecture, Professor Iqbal. I'm interested with your research about antimicrobial because it is somewhat similar with my research. I want to ask you about the best way to develop such antimicrobial compounds to be oral drug instead of topical application. In your opinion, what's the best model in animal that can be used for this purpose? Okay, please.
Okay, Professor Andre, this bacteria, this so-called infectious model are the easiest to develop. That in among all the models, uh, the best models which we can develop for topical application, of course, you need to have, if you are developing a topical anti-infectious agent, you can always develop topical infection. So you need to immune compromise because often these rodents do not really, they are so hard that it's very difficult to develop a topical infection on them, and you need to immune compromise them. So you need to give them some amino suppressant, and then only you would be able to develop candida infection or staphylococcus aureus infection or pseudomonas infection. So topical application, topical infectious models are very, very easy, but you need to suppress the immunity of the animals also. For systemic model, which is required for oral or injectable antimicrobial, then it's called bacterial load, so-called microbial load model, in which you inject bacterial cell in certain numbers, and then you measure the bacterial load by using flow cytometry. And this is a time course study, because with time you would be able to see that the bacterial cells multiply, you have more of it, and often you may not really see any infection. I mean, you may see lots of bacteria in the blood, but you may not see any shivering or any fever or anything like this. This is typical of animals. Often they do not develop symptoms, but they have lots of bacterial. So a bacterial load, so-called bacterial load, is one model through which you can see whether your compound is capable of decreasing the bacterial load in a dose-dependent manner. Is exciting, but of course, always remember that in the field of anti-infectious diseases, the work always start from in vitro screening, 96-well screening, in which you have standard cultures, because that is the beginning of the work, and only then, when you see the activity demonstrated in vitro, then you go to the animal model.
Okay, Pak Abdi. Okay, thank you very much for explanation, Professor Chaudhary. Your presentation is quite interesting. Still more two questions in the chatting room. Okay, next question come from Ibu Sofa Fajria from Indonesian Institute of Science. The question is, thank you very much for nice presentation, Prof. Chaudhary, regarding integrating natural and scientific approach. Usually in natural product, we only got a little amount of compounds, especially for new compounds. Could you give us suggestion how to get efficient of isolation or scientific of natural products? Thank you very much.
Okay, I agree with you. It is a problem, and that is a problem which would always be there, because natural products are often, secondary metabolites are often available in very little quantities. So I honestly, I do not have a solution, because if you harvest plants from the wild, and then you try to isolate compound from that, you literally destroy forests. So I would not recommend that you go to collect a plant from the wild and then try to isolate large quantities of natural product. The best is ex situ cultivation. If you can carry out ex situ cultivation, a plant can be grown it can be cultivated out of wild. That is one source, and that the Chinese are trying to do that also, because the Chinese are growing many of these medicinal plant ex situ. So this remain a problem. Second is, of course, you can produce them in enough quantities if you develop alternative methods. One, as I mentioned, metabolic engineering. It's possible to identify the gene of enzymes which are responsible of catalyzing the production of that, and then modify those genetic engineering by using genetic engineering and produce them in large quantities by E. coli, Saccharomyces. Many of the natural products have been produced. The third way is biomass. So you produce these natural products in cell culture. Instead of getting them from the wild, you can grow them in bioreactors. But having said that, this is remain a problem. I would advise you to read a very interesting paper on the production of Taxol. The similar problem was faced when Taxol was identified as an anti-cancer drug, and they wanted to have only 10 grams of that, and they could not produce because Taxol is in the bark of Taxus brevifolia and need to beat the bark of a tree to produce them enough quantities, semi-synthetic or fully synthetic.
Okay, Ibu Sofa.
Thank you very much, Prof. Chaudhary. Nice to meet you in here again.
Thank you.
Yeah. Okay. Thank you, Prof. Andrea.
Okay, thank you very much. You're welcome. Okay, we move to next question from Dr. Andri Frediansa from Indonesian Institute of Science. The question is, thank you for our some presentation of Professor Chaudhary. What do you think about the discovery of natural bacterial product at this time or recently? Why most people rarely find the new or novel structure or even new natural product classes, even though genome mining is starting increasing? This evidence could be seen in the Journal of Antibiotic published by Nature. The most researchers only found derivatives or analog. During the antibiotic era, we knew that many antibiotics had been discovered from bacteria. How about in Pakistan, related microbial natural products? Please.
Okay, again, a very interesting question. You see, most of the anti-infectious agent, antimicrobial agents which we use today are of natural origin. So it's very obvious that majority of the natural product, majority of anti-infectious agents which we see use today, either from bacterial sources or from fungal sources or from plant sources. And so they remain a very important source of it. Problem with the antibiotic is that not many people are working in the field also now. Pharmaceutical companies do not work in the field of anti-infections, because of a very simple reason. They spend $2 billion, $2.5 billion. They know that within few years the resistance is developed, and often before they get return of the investment, their antibiotics is already useless. So not many pharmaceutical companies are working in the field of antimicrobial drug discovery. In Pakistan, we in our institute we are working on the discovery of antimicrobial agents, but not antimicrobial agents sole. We have now a separate laboratory for resistance reversal agents. So what we are trying to do is the following, that we're trying to identify compounds which help existing antibiotics to function. We consider this approach is a very interesting approach because development of antibiotic from the scratch is expensive. Augmentin is a one classical example in which antibiotics is supported by a beta-lactamase inhibitor. So it's possible to develop antimicrobial formulation by using existing antibiotics and resistance reversal agents. And that resistance reversal agents are often you can obtain from the natural sources. Now, again, how much you can obtain is a problem itself. But my response to your good question is that this is an extremely important area. This is an area on which human survival based. Resistance of microorganisms is a great challenge for all of us, and we all must work in it. Pharmaceutical companies are not interested, but at least academic institutions should continue to contribute in this field.
Okay, it means there is an economical aspect, not so interest for some companies to develop the new antibiotic.
Absolutely.
Okay. Okay, there is no question in the chat rooms, but if possible, I will have one simple question, Professor.
Please.
Okay, this is only a simple question. During your long career as scientist, which research very special for you? Or maybe your research result special or something, and why you think that is special? Okay, please.
You see, I've been working in this field since last 100 years. You know, I'm just so old now. But and it really excites me even today also. When I isolate a new compound, when my student brings something new for me, I feel the same excitement as I used to feel centuries ago. But one research which I feel I have greatest satisfaction is the one which I have done with the skin diseases. You know, when you will come to Pakistan, I'll show you that certain regions of the country, there are a tropical disease called leishmaniasis. It's also in Indonesia. It's very, very common disease, but in Pakistan is unfortunately very endemic, and many regions you find this sandfly producing, transferring this vector, and people have lots of patients, and often they live with it for the rest of their life. It's not fatal. It doesn't kill a person, but it really have a problem, especially for young girls, because I've seen young, beautiful girls with scars on their face, their noses, and I really felt bad about it. Now, what we have done was absolutely a very simple science, nothing exciting. What we have done was that we asked them what they use, and then, you know, out of this ethnobotanic survey, we identified that in some region of the country, people have been using simple natural product for the treatment, and sometimes they were able to treat that also. And on the basis of those ethnobotanic formulation, we developed a natural product-based ointment, which is now available in the country for leishmaniasis. And over 2,000 patients, clinical trials were conducted on 2,000 patients, because this is a very common disease. So a very simple science gave me lots of satisfaction. Of course, I've done lots of complex sciences. I have many studies conducted at a molecular level. But at the end of the day, what you can see happening to the people, and if you can help them in some way, ultimately that gives you the greatest satisfaction.
Okay, thank you very much, Professor Chaudhary. Yeah, I met 13 years ago with Professor Chaudhary in Bangkok, and Professor Chaudhary asked me to move to Pakistan, but I don't know what the reason not reliable for me at the time. Okay, thank you very much, Professor Chaudhary. And also time is over. I would like to say thank you very much for all keynote speaker, Professor Gunawan, Dr. Pradipta, and also Professor Chaudhary. Thank you very much. Okay,
thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Okay, I return back time to the organizing committee. Ibu Marisa, please.
Thank you, Professor Dr. Andrea Agusta, for the nice presentation today. Dear our speakers and our moderators, and also all of the participants, thank you for the time with a nice presentation today and also fruitful discussions. We will have a break time from 12 until 1 p.m. But before break time, I want to tell about our next schedule. Next at noon, we have 2 parallel sessions. The first parallel session will be start from 1 until 3 p.m. The first parallel session will be divided into 3 rooms. Room A, about natural product and traditional medicine, will be moderated by Dr. Sofa Pajria. The second room will be discussion about pharmaceutical technology, drug delivery system, analytical chemistry, and chemical engineering with the moderator, Siti Nurul Aisyah Jenny, PhD. And the third room will be discussed about food technology and functional food with the moderator Hakiki Melanie, PhD. Ladies and gentlemen, the second panel session will be start from 3:15 p.m. until 5 p.m. and also will be divided into 3 rooms. The first room about natural product and traditional medicines and will be moderated by Abdi Wiraseptama, PhD. The second room will be discussed about analytical chemistry, chemical engineering, and will be moderated by Dr. Indri Badriah Adilina, Master of Engineering. And the third room will be discussed about food technology and functional food and will be moderated by Khairun Nisa, PhD. We hope all the participants will be joined in each room until end of this day. And the participant will automatically enter each room according to the schedule that you can read in the program book. Okay, we hope all of the participants enjoy the symposium. See you tomorrow. Bye-bye. Assalamualaikum warahmatullahi wabarakatuh.
Kita boleh live ya?
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. This is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food. Yogyakarta. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned
as
foods.
a sterilizing agent.
After
Then
the
in the filling
sterilization
step,
process,
gudeg are filled
canned
in
food
cans.
products are cooled down at room temperature
The
and
next
then
step
quarantined
then
for 2
continued
weeks
by
in sterile
exhausting
room
and seaming
for quality
process.
controlling. The cans are labeled and ready to be sold.
After being tightly sealed, the cans go under another sterilization step using
The
autoclave.
implementation of the technology
This
is
sterilization
able to
process
enhance the
is an
production
essential step
capacity
for
of
the canned
foods
foods.
and increase the sales turnover
After the
in
sterilization
Yogyakarta's
process,
traditional food
canned food
processing
products are
industries.
cooled down at room temperature and then
Furthermore,
quarantined for 2
these
weeks in
culinary
sterile room
industries
for
are getting
quality
more
controlling.
knowledge on the hygiene,
The cans are
safety,
labeled and
and
ready to be sold.
standardization of food processing technology. The packaging technology has received a certification of good manufacturing
The
process,
implementation of
or
the technology
GMP,
is
from
able
the
to enhance
Indonesian
the
National
production
Agency
capacity
of
of
Drug
canned
and Food
foods
Control.
and increase
In
the sales
addition,
turnover
canned
in
traditional foods
Yogyakarta's
have
traditional
received
foods
positive
processing
responses from many
industries.
people.
Furthermore,
Yogyakarta
these
will
culinary
always be the
industries
city
are
that
getting
feels
more
like
knowledge
home.
on the hygiene,
However,
safety,
it's
and
always been nice to be
standardization
back to
of food
Yogyakarta,
processing technology.
the city of
The
yearning
packaging
with a
technology
greatly
has
delicious
received a
traditional
certification
local
of
food.
good manufacturing process, or GMP, from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with a greatly delicious traditional local food.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan,
Yogyakarta,
angkringan, home
the
industry,
city of education and
restaurant.
culture, has its own stories and
But then,
romances
how
in
can
every
we fulfill
corner of the
our
city.
longing for these traditional
Each
foods
corner will
when we
bring
are
us
far
back to the
away?
memories that make us to always come
Research
back
and
to the
Development
city of
Division
yearning.
for Natural Materials Technology,
Yogyakarta has
Indonesian
some traditional
Institute of
foods,
Sciences,
such
has
as
been
gudeg,
developing packaging
mangut lele,
technology for
lombok
ready-to-eat
ijo,
foods.
tempe bacem,
Traditional
and
foods are
brongkos,
processed
which
and
are findable
packaged
in
in
street food
cylindrical
stall,
can, so
lesehan,
it has longer
angkringan,
period of shelf
home
life
industry,
and
and
increase
restaurant.
its value. The implementation of
But
packaging
then,
technology
how can we fulfill
developed
our longing
consistently
for
in
these
the
traditional
institution.
foods when we are working
In
along with
Javanese.
partners to
So,
implement the
traditional
packaging
food
technology?
produced was then called as
They
gudeg
managed to
by
process
people
and
of
package
Yogyakarta.
42 kinds of traditional foods from
Gudeg
all
is
over
served with
Indonesia,
addition side dishes
such as
pindang
mangut lele,
egg,
lombok
slices
ijo,
of chicken meat,
tempe bacem,
krecek condiment,
tempe kari,
tolo beans,
brongkos.
tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in
Was
autoclave.
then called as
An
gudeg
autoclave
by people of
used
Yogyakarta.
high-pressured, high-temperature steam
Gudeg
as a
is served with
sterilizing
addition
agent.
side dishes such as
Then in
pindang
the
egg,
filling step,
slices of
gudeg are
chicken meat,
filled in
krecek
cans.
condiment, tolo beans, tofu.
The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food. Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817 with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research, social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research, scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages flora-fauna collections. The Bogoriense Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers, 2. Accrediting scientific journals, 3. Studying and evaluating biodiversity, establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia Pacific Centers for Eco-Hydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future.
Yogyakarta has some traditional foods, such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. Manufacturing process, or GMP, from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817 with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, and electronics, eco-friendly transportation, advanced materials and nanotechnology research, social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research, scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the third largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers, 2. Accrediting scientific journals, 3. Studying and evaluating biodiversity, establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia Pacific Centers for Ecohydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significant. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods, such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia with the coordination of the Ministry of Research, Technology, and Higher Education.
Halo, Assalamualaikum warahmatullahi wabarakatuh. We symposium. Okay, the honorable speakers, distinguished guests, ladies and gentlemen. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to welcome you all to this prestigious seminar, the 6th International Symposium of Applied Chemistry Webinar Conference, especially in the natural medicine and nutraceutical topics. In this occasion, we shall have outstanding presentations from our prominent presenters, whom I believe shall enrich our insight and knowledge regarding the themes of this seminar. Before the presentation begins, allow me to introduce the presenters. We have 8 presenters divided to sections. Every section has 4 presenters, begins with presentation from each speaker, and continue question and answer after the 4th speaker presentation. As information or chat to ask your question. Okay, we start the first section. Our first presenter is Ibu Sri Priyatni, continue with Ibu Linda Sukmarini, Bapak Gunawan Pasaribu, and Bapak Ahmad Nandi. Without further ado, I would like to welcome our first presenter, training of anti-diabetic activity and the cultivation study of local marine microalgae. Ladies and gentlemen, please welcome Ibu Sri Priyatni. The floor is yours.
Sorry.
Sorry. Matt, is it problem in there? I think the voice not clear. Maybe you can present directly, this Priyatni. Have you on your slideshow?
Yes, I have.
Okay, please, Pak Hikmat can stop the screen and Ibu Priyatni will use his screen. Halo, Pak Hikmat. Can you hear me? Or Mbak Yani? Because the voice from the screen cannot hear.
Bu Sofa, just a minute, okay?
Okay.
Just a minute. Okay, we will check the connection. Okay.
Okay. Thank you. Thank you. Microalgae with potential as antidiabetic sources that can be applied in. Okay, now for the first I will give thanks to the organizer and chemistry. Okay, all participants, we going to the first presenter, Ibu Sri Priyatni. Please welcome Ibu Sri Priyatni will be presentation study of local microalgae. Please for Ibu Sri Priyatni, the floor is yours. Hello, Pak Hikmat. Can you continue the presentation next? Or maybe Dr. Sri Priyatni will be presented by his screening. Hello. Okay, Ibu Sri Priyatni, could you explain your PPT? The time is about 10 minutes for you. Okay, Ibu Sri Priyatni, please come to deliver your presentation and entitled with the screening of anti-diabetic activity and the cultivation study of local marine microalgae. Ladies and gentlemen, please welcome Ibu Sri Priyatni. The floor is yours.
Okay, can you see the slide?
Yes, I see. Okay, please start, Ibu Sri Priyatni.
Okay, thank you for the organizing committee for the presentation in the ISAC. In this occasion, I would like to present my farm. Also use the lamp, white lamp at about 500 to 1,000 lux. And the second method, we use the cycle incubator. To counting the cell number of microalgae that growth, we use the cell counter on 100. So the cell number can calculate according this formula. And then the biomass extraction, we use methanol solvent with the ratio 1 to 20, conducted at 60 degree, 100 p.m. for 2 hours. The methanol extract, then we centrifuge and evaporated to have the concentrate of extract. And for the IPS or intracellular polysaccharide extraction, we use only the distillated water and heat it at about 95 degree for 6 hours. Then after that we fill and the flow.
Peptide-derived marine natural products are a class of compounds with diverse and interesting biological properties of mainly pharmaceutical interest, such as antimicrobial, antiviral, anti-cancer, cytotoxic, and anti-allergenic or analgesic. In this presentation, we will focus on marine peptide compounds in advanced drug development for clinical use and pharmaceutical application. Marine peptides exhibit remarkable diversity both in their structure and biological activity due to the versatility of their biosynthetic machinery. They are mainly synthesized either by ribosomal machinery or by non-ribosomal pathway. Ribosomal peptide oryx depend on a set of the standard 20 canonical or proteinogenic amino acids, in which diversity is generated by post-translational modification. On the other hand, non-ribosomal peptides are synthesized by a large pool of proteinogenic and non-proteinogenic amino acids by a large multimodular enzyme complex of non-ribosomal assembly. Moreover, non-ribosomal peptides are sometimes occur as hybrid with polypeptides. To keep in mind, most of the marine reefs and non-ribosomal peptide drugs have been the subject of two structure-activity relationship studies and in consequence have reached the market as semi-synthetic or synthetic compounds in multiple variations. Now, let's have a look into the development of reef drugs and NRPS drugs. A commercial reef drug and the first US FDA approved marine drug is chiconotide, which is now known under the trade name Berdaulat, for the treatment of chronic pain. Chiconotide is a synthetic analog of conopeptide omega-conotoxin MP2A. The naturally occurring of omega-conotoxin is isolated from marine conch snails, Conus malvis. The analgesic property of Berdaulat is attributed to its ability to block specific voltage-activated calcium channels in neurons. Moreover, other modified conotoxin such as contotoxin G and X-conotoxin MA1 so far unfortunately have been discontinued due to the side effect toxicity when they reach phase 1 or clinical development, including the hepatocyte cyclodextrin trial for COVID-19 treatment following the successful in vivo study and phase 2 trial. In addition, other CDP is elisitepsin or Ipvalec, also developed by PharmaMar, but this peptide drug was filed in phase 2 due to the safety concern. In addition to CDP pitidepsin, these are some examples of non-ribosomal peptide hybrid drugs that already been approved for the treatment of refractory soft tissue sarcomas. The analog of trabectedin called lurbinectedin under the trade name Zepzelca has also been approved for the treatment of older patients with metastatic small cell lung cancer. And this drug is already in the market in the US since last July this year. Another example is an antibody drug conjugate NRPS called brentuximab vedotin under the trademark Adcetris. This drug has been approved for the treatment of Hodgkin lymphoma. And interestingly, there is a drug as protease inhibitor for anti-cancer agent, namely marizolmib or salinosporamib A, isolated from Actinomycete salinospora tropica. And this peptide drug is currently under phase 3 trial in humans. To recap, marine peptide has shown fascinating structural diversity and also remarkable biological property, as many of them are successful in the market. However, there are still challenges in supply shortage and synthetic difficulties. Therefore, the progress in advancement technology, especially in biosynthetic pathway engineering, would help to design newly peptides with biological and pharmacological properties. This brings to the end of this presentation. I would like to thank you for your attention and time. Thank you.
Thank you very much for Linda Sukmarini for a nice presentation about drug development from peptides derivative with marine natural products. Okay. We can say we can build once again a big round of applause for Ibu Linda Sukmarini for her nice presentation. Now, ladies and gentlemen, welcome our third presenter. He will deliver a presentation with the title Toxicity and Total Phenolic Content of Sauria Vulcani from Cultivation. Ladies and gentlemen, please welcome Bapak Gunawan. The floor is yours.
Good afternoon, everyone. I'm Gunawan Pasaribu from Forest Product Research and Development Center. Now, I'm PhD candidate at Universitas of Indonesia. Today, I'm going to talk about the toxicity and total phenolic content of Sauria vulcani extract from cultivation. I'm Gunawan Pasaribu with Sumudiono, Budianto, Antonius Hari Cahyana, and Ustadz Bari as the author. Indonesia is named as mega biodiversity, rich of flora and fauna. There are so many plant resources for many applications, such as for food and feeds, pesticides, cosmetic, additives, spices, and medicine. On the other hand, it is few number of plants have been applied commercially. So we need to explore or bioprospecting of bioactive of plant extract and their active compound. The object of my research is the ethyl acetate and methanol Sauria vulcani extract. The picture below, let's see that the plant of Sauria vulcani, the first one show the leaf and the flower, and the second one show the cultivated Sauria vulcani in the rehabilitation program by the Ministry of Environment and Forestry. In practical, Sauria plant usually used while plant as research object and in utilization. Different with this research, that material plant used is the cultivation at the age of 4 years. So we hope in this research we'll investigate how the different in secondary metabolite produced from one plant and cultivated plant. There are some chemical compounds from Sauria sp. We see that generally the compound is in terpenate group. It is the bioactivity of genera of Sauria sp that I collected. The first one is antioxidant activity, anti-cholesterol, anti-diabetic activity, anti-hyperlipidemic, analgesic activity, antimicrobial activity, wound healing activity, and immunostimulant activity. There are the chemical compound of Sauria vulcani. So the three of chemical compound is the triterpenoid group. This is the, and then we powder with hammer or blender, and then we extract using maceration technique with multi-level extraction using enhexan, ethyl acetate, and methanol solvent. And then filtrate, we concentrate using a rotary vacuum evaporator for extract yield. This is the phytochemical testing based on carbon. It is include alkaloid, flavonoid, saponin, tannin, triterpenoid, and hydroquinone. The toxicity, we use the brain lethality test method using the Artemia salina egg. The mortality is the terminine, and the LCVT was calculated by probit analysis. Total panel content based on follicular TAC. It is used lambda max in 765. The phenolic content in the extract is expressed as milligram of gallic acid equivalent. This is the result and discussion. The first one is the phytochemical. The phytochemical show the presence of flavonoid, steroid, tannin, and saponin in hexane, ethyl acetate, and methanol solvent. The toxicity testing show LC50 value and hexane 365, ethyl acetate 750, and methanol extract is 135 ppm. Toxicity value is classified as non-toxic if the LC50 more than 1000 ppm. The low toxic is classified as the LC50 from 500 to 1000 ppm. The medium toxic is classified if the LC50 from 100 to 500 ppm. The high toxic is classified as the LC50 from 0 to 100 ppm. This is the total panel content. So N-hexane is 7.1, ethyl acetate is 13.7, and the methanol is 16.5 gallic acid equivalent. This value indicate that in every gram of extract of N-hexane, ethyl acetate, and methanol, equivalent of 7.1, 13.7, and 16.5 milligram of gallic acid. When compared with the result of the total phenolic content in wild and cultivated Sauria vulcani, it shows different result. Total phenolic content of plant from cultivation is tendency smaller than from wild. This is probably influenced by the age of the plant, which is still quite young. It is 4 years. In general, the Sauria vulcani plant taken from nature. It is the mature and bigger. This is the last of the summer. The first one, the hexane and methanol-sauria volcanic extract were classified as moderate toxic, while the ethyl acetate extract was classified as low toxic. The second one, the total phenolic content of hexane, ethyl acetate, and methanol extract were 7.1, 13.7 and 16.5 milligram malic acid equivalent respectively. This is the end of my presentation. Thank you.
Thank you very much for Bapak Gunawan Fasidu for my presentation about the toxicity and total phenolic content of soda volcanic extract from cultivation. Before we going to the fourth presenter, if you want to give question, could you chat from you can write in the chat or raise your hand. Thank you. So let's give once again a big round of applause for Bapak Gunawan. Now, ladies and gentlemen, let's welcome our fourth presenter. He will deliver a presentation with entitled In Vitro Lipid Lowering Effects of Kalianda Caloterus Extract. Ladies and gentlemen, please welcome Bapak Ahmad Randi. The floor is yours.
Okay, good afternoon, ladies and gentlemen. First of all, I would like to thank to the ISAC 2020 Organizing Committee for giving me the chance to present our research result. And for this occasion, I would like to try to present our research result. The title of this presentation or this research is In Vitro Lipid Lowering Effects of Kalianda Caloterus Extract. This research was conducted by myself, Ahmad Randi, from the Research Center for Chemistry, Indonesian Institute of Science, Kim Yong-sok, and No Juwon from the Korea Institute of Science and Technology. So basically, in this research, we would like to find a new strategy or new alternative based on natural product to improve the fatty liver disease condition, especially non-alcoholic fatty liver disease, or here I will mention it as NAFLD, non-alcoholic fatty liver disease. So here we can see in this above picture there are several factors that can initiate fatty liver disease, but we would like to focus on one initial factor, which is diet visceral obesity or excessive lipid accumulation induced by a high lipid diet that can induce obesity. So fatty liver disease, which is alcoholic or non-alcoholic, is an excessive fatty liver or lipid accumulation in the liver, and it can trigger several degree of severe liver condition. In this below picture, we can see the healthy liver, and when it is when there is a excessive lipid accumulation in the liver, it can trigger lipogenesis and then steatosis. And chronically, if the condition is become more severe, steatosis can induce further inflammation, and it can trigger to NASH or non-alcoholic steatohepatitis, and it also can trigger oncogenesis or liver cancer. And the end result of this oncogenesis is the dysfunctionality of normal liver cell. In this case, the condition can become more severe to become a cirrhosis, where the normal liver function is not working well, and the normal liver cell is converted to scar tissue. Here we can see that beside the lipid accumulation in the liver, high-fat diet in case of obese patient and also affect the white adipose tissue. We can see here that high-fat diet can trigger the increasing number of white adipose and also increasing lipid accumulation in the white adipose. in the effect of increasing lipid accumulation in the white adipose tissue, there will be increasing free fatty acid influx to the liver, and it also can trigger lipid accumulation in the liver that can result to steatosis and also fatty liver disease. So the plant material that we study in this research is Calliandra calypsis. We study the ethanol extract of Calliandra calypsis leaf. So basically, we already conducted several screenings from hundreds plant extract, and we found that Calliandra calypsis extract, or CCE, is one of the most potent plant extract that can lower the lipid accumulation in our study model. So the objectives of this research is to investigate the potency of CCE, or Calliandra calypsis extract, to improve obesity-induced NAFLD, non-alcoholic fatty liver disease, by attenuating fatty acid-induced lipid accumulation in vitro in FG2 cells model, and also inhibiting adipocyte differentiation in 3D3L1 cells. Here I highlight several key keywords. CCE is Calliandra calypsis extract, and we use obesity in this NAFLD model to induce lipid accumulation in vitro in FG2 cell and 3D3L1. So this is our methods. We use two in vitro model. The first one is we use FG2 to model the liver cells, and we also use 3D3L1 to model the white adipose cell, or white adipose tissues. In FG2 cell, we observe the lipid accumulation by lipid staining, or redox staining, and we also observe the important factors or protein or enzyme in the fatty acid synthesis and also fatty acid oxidation by using Western blot and reverse transcriptase PCR, and we also observe the level of AMPK, which is an important factor or enzyme in the lipid metabolism. We observe the activation of AMPK by Western blot. And when using 3D3L1 white adipose, we observe the adipocyte differentiation, and we analyze the lipid accumulation by redox staining, and also the factors affecting the adipocyte differentiation, which are PPAR gamma, CBP alpha, and CBP beta by Western blot. And this is the result. We show that CCE, or Calliandra calypsis extract, inhibits lipid accumulation in FG2 cells. Here we use the lipid accumulation model by inducing lipid accumulation by palmitic acid in FG2 cells, and we found that treating the FG2 cell with Calliandra calypsis extract lowers the lipid accumulation compared to the group without CCE treatment with only palmitic acid. And we would like to observe whether this lowering lipid accumulation was conducted by CCE by increasing beta oxidation. We observe the PPAR alpha, CPT1 alpha, and also PGC1 alpha. Here we can see the beta oxidation, or lipid oxidation metabolism in the liver cell, where fatty acid is converted to acetyl-CoA, and the important factors here are PPAR alpha and PGC1 alpha, which can trigger or activate the expression of CPT1, which is an important enzyme in the fatty acid oxidation. In our result, we show that the level of CPT1, PPAR alpha, and also PGC1 alpha is increasing, and the result is the increasing rate of beta oxidation and increasing rate of fatty acid breakdown to acetyl-CoA. Meanwhile, we also observe the important factor or enzyme in the lipid biosynthesis. Here we can see the de novo synthesis of acetyl-CoA synthesis, acetyl-CoA conversion to fatty acid. Here there are ACC or acetyl-CoA carboxylase, and also FAS or fatty acid synthase that are important enzyme in the conversion of acetyl-CoA to fatty acid. And these two enzyme level were regulated by SRBP. Here we can show that the treatment of CT2 cell with CCE will lower the level of SRBP, ACC, and also FAS. And the end result of this CCE treatment is inactivation of fatty acid synthesis. We also show that the MPK is activated, the level of phosphorylated MPK is increasing by treatment of CCE, and the end result of this MPK activation is a phosphorylation of ACC. When the ACC is phosphorylated, it becomes inactive and it cannot convert acetyl-CoA to fatty acid. So the result of activated AMPK pathway is the lower lipid accumulation by blocking the lipid biosynthesis pathway. In the 333L1 model, we use 333L1 to model the white adipose tissue differentiation. We treat 333L1 with IBMX, dexamethasone, and insulin to induce the adipocyte differentiation and in hope to increase the lipid accumulation. And here we show that the treatment of 333L1 with CCE inhibit the lipid accumulation compared to group that were not treated with CCE. And we would like to observe whether the lowering lipid accumulation in 333L1 is by inhibiting the adipocyte differentiation. We analyze the level of PPAR alpha, CEBP alpha, and CEBP beta. These are an important transcription factor that can induce the adipocyte differentiation. And we show that treatment of 333L1 with CCE can reduce the number of PPAR alpha, CEBP alpha, and also CEBP beta. So we propose that treatment of CCE to the adipocyte can inhibit the adipocyte differentiation and also inhibit the lipid accumulation. So in conclusion, from this in vitro study, we show that the CCE or Kalian glycolysis extract can lower lipid accumulation to three mechanisms. The first one is attenuation of lipid accumulation through regulation of lipid metabolism in the T2 cell. The CCE can increase the beta oxidation and also can inhibit the fatty acid synthesis, and the end result is lowering lipid accumulation. And also the CCE can activate AMPK in the T2 cells that can trigger the increasing rate of fatty acid oxidation. Meanwhile, the CCE can also attenuate adipocyte differentiation in 333L1 adipocyte model. So in conclusion, overall aspect of CCE is the lowering level of lipid accumulation in both liver cell model and white adipocyte tissue model. So I would like to thank to the University of Science and Technology and KCM Institute of Natural Product to enable this research. And also I will welcome any question, suggestion, or discussion in this conference. Or you can email me at this email address ahmad.randi@lipi.go.id so we can have further discussion. So thank you for your attention.
Thank you very much, Dr. Ahmad Randi, for nice presentation. So we can let's give once again a big round of applause for Bapak Ahmad Randi. Thank you very much for all presenters for the informative and interesting talks. Very well, ladies and gentlemen. Now we come to question and answer questions. From chat, we can two questions for Ibu Sri Priyatni. Maybe all participants can use right hand directly to the speaker, or you can chat now. Maybe about 7 minutes. Again, we have a time. For Ibu Sri, from Ibu Muhani Unti, she asked about how many the ICCP value of the compound which is potential to be anti-diabetic. Maybe I can also, you have two questions from Ibu Muhani and Ahmad Fadli. Maybe I directly answer for Ahmad Fatoni. Microalgae as chlorella in other studies shown as anti-diabetic due to its metabolite, for example, carotenoid of astaxanthin and lutein. This metabolite has antioxidant activity as well as anti-diabetic activity. Could you tell me the metabolite related to the anti-diabetic activity in your study? Please, Ibu Sri, could you answer these two questions? Thank you.
Thank you for the moderator. I would like to share the data about the anti-diabetic activity. So from the screening, we can see the anti-diabetic activity from the positive from the methanol extracts. Is the maximum is 12, the inhibition is 12.63 for the porphyridium. And for the second is from chlorella. This is we use control acarbose is about 91. So if you want to have higher than this activity, so we should the purification the metabolite, and we can have the high activity like this control acarbose. But usually for the natural product, we can use this directly when we find the positive for anti-diabetic activity. It's my opinion. So we should continue the study to purification the metabolite, something like that. And to answer the second question, there's a metabolite from chlorella, like you write in the question, yeah. Also from other microalgae, there's another metabolite depend on the microalgae. In this research, the porphyridium is the highest. They contain picobiliprotein is the highest metabolite in the porphyridium. As the research other study from my study, porphyridium is contain picobiliprotein like red pigment. Porphyridium have a red pigment, and the majority is picobiliprotein. I guess that's metabolite that contribute to the anti-diabetic activity. Maybe like that, Bu Sofa.
Okay, thank you, Ibu Sripriyatni. Maybe any comment from Ibu Muhani and Bapak Ahmad Fatoni for the answer?
Okay, thank you very much for your answer.
Okay, Ibu Muhani, is it clear, Ibu Sripriyatni, the answer of Ibu Sripriyatni? Okay, thank you. Maybe we have 3 minutes question from participants. Okay, I just want to ask to Ibu Linda. It's very interesting about drug development from peptide that is marine natural products. Yeah. How the implementation? Yeah, how the implementation? Because you in your slide that you collaborated with Pharma, is right? So how implementation to the next as drug development from marine natural products? Thank you.
Okay, so your question is about Aplidin, ya?
Yeah.
So far as I know, it's last October. They tried to, in the last May, as far as I know, they tried to initiate the phase 1 clinical trial for COVID-19, and then afterward they got some good result, and then on the end of October, they already prepare for the next phase 3 clinical trial. So I think they are now in preparation now. Hopefully, so it's anti-drugs,
ongoing.
It's ongoing, yeah.
Okay, thank you, Ibu Linda. Now we have a question from Ibu Elvita from Universitas Wijaya to Bapak Gunawan Pasaribu. The question is, how is the correlation between total knowledge and the toxicity or ICCT? Please, Bapak Gunawan Pasaribu, could you answer the question from Ibu Elvita? Thank you.
Okay, thank you, Bu Elvita. It has a linear correlation. The higher toxicity, it is the higher total panel too. That's all. Thank you.
How Ibu Elvita is it feel the answer from Bapak Gunawan? Okay, I think it is clear, Bapak Gunawan. There is we have correlation between knowledge and toxicity, right? Okay. Maybe I have a question for Bapak Randi. Is it your study can how to implementation in the future for Bapak Randi to the lipid lowering itu from kalianda kalau tripus extra?
Thank you, Bu Sofa. Our maybe liver disease is quite difficult to cure if the liver disease is already very severe. So the implementation of this research, perhaps it can be, if the natural product is edible, so it can be given is in the form of functional food. In case the liver disease is not start to become severe, so when it is maybe we can think the implementation as a functional food, not as a medicinal drug. Maybe that's my opinion. Thank you.
Okay, thank you very much, Bapak Randi. Thank you very much. And this is the end of the first section. Thank you very much for Ibu Sri Priyatni, Ibu Linda Sukmarini, Bapak Gunawan Pasaribu, and Bapak Randi. We can give big applause for them. Okay, ladies and gentlemen, we're going to the next session. We have 4 speakers. Maybe we can, first is Ibu Lucia, continue with Bapak Fahrauf, and maybe could you have, maybe in the list, Bapak Muhammad Yanis Musja, in here? Bapak Yanis? Okay, Bapak Yanis, and the last is Ibu Sylvia. Bapak Yanis didn't join. Okay, we're going to the next session. We have 3 speakers now, Ibu Lucia, Bapak Fahrauf, and Ibu Sylvia. Without further ado, I would like to welcome our first presenter in the section 2, Ibu Lucia Dwi Antika, will be a presentation with the entitled Anti-Osteoclastogenic Activity of Glycochalcone, antagonizing rank RAMKL mediated bond resorption in murine macrophages. Please, for Ibu Lucia Dwi Antika, the floor is yours. Thank you.
Good afternoon, everyone. My name is Lucia Duyantika from Research Center for Chemistry, Indonesian Institute of Sciences. Today, I'm going to present our study entitled Anti-Osteoclastogenic Activity of Liposarcoma A, Antagonizing Runckel-Mediated Bone Resorption in Urine Molecules on Postmenopausal Older Women Due to the Estrogen Deficiency-Related Menopause and Type 2 Senile Osteoporosis. It is an age-related predominance of bone loss. Usually occurs in elderly due to the impaired bone remodeling process. Due to the prevalence worldwide, it is reported that over 200 million people suffer from this disease. And then by 2050, it is expected that the hip fracture incidence will be increased by 240% in women and 310 in men. Previously, I mentioned about bone remodeling. Bone remodeling is a continuous cycle of the removal of old bone by osteoclasts and deposition of new bone by osteoblasts. In the initiation of bone remodeling, RANK ligand and OPG, its natural inhibitor, were released by osteoblasts. The RANK ligand bind to RANK receptor on osteoblast precursor, which initiates the cell fusion and then formation of multinucleated osteoclasts. And osteoclasts attach themselves to the surface of the bone and being activated. The osteoclast activated and form the special structure called actin ring. And then after that, the osteoclast release acid and lysosomal enzyme to dissolve bone matrix and complete the process of resorption. Under some circumstances, bone is broken down much faster than it can be renewed due to the bone loss disease, including osteoporosis. In this study, we use lipocalcone A as a sample. Lipocalcone A is a dietary flavonoid from the root of Glycyrrhiza inflata and Glycyrrhiza glabra that has been reported to possess various pharmacological activities, including anti-inflammatory, anti-angiogenic, antioxidants, and antibacterials. In this study, the effect of lipocalcone A in cell viability was measured by MTP colorimetric assay. It was found that lipocalcone until concentration of 20 micromolar show no toxicity against the cell culture. The purpose of this study is to determine the potential mechanism of lipocalcone A against Runckel-induced osteoclastogenesis in vitro. In vitro study was conducted using the raw murine macrophage cell as a pre-osteoclast model. The cells were treated with Runckel in the absence and presence of lipocalcone A, and then the medium was changed every 2 days. The cells were lysed, and in the day 5, the cell lysate was subjected to SDH and Western blot analysis with specific antibodies. Natural osteoclasts were characterized by having 3 or more multinuclei and secretes the TRAP enzymes. So, in this study, the TRAP staining was performed as a marker for mature osteoclasts. You can see the picture. These results show that lipocalcone reduced the number of TRAP-positive multinucleated osteoclasts, and TRAP enhanced by Runckel was decreased in lipocalcone-administered cells dose-dependently. Mature osteoclasts dissolve bone matrix by releasing lysosomal enzyme and acid to dissolve the bone matrix. To examine the effect of lipocalcone on bone-resorbing activity of osteoclasts, the Runckel differentiated row cells were cultured on calcium phosphate plates and treated with lipocalcone. Mature osteoclasts created Zebral's resorption pit. Meanwhile, lipocalcone efficiently reduced the pit area. Osteoclast activity was evaluated by measuring the fluorescence intensity in the culture medium. And then the results show that treatment with lipocalcone A decreased the fluorescence intensity dose dependently. Previously, I mentioned about the actin ring cytoskeleton. Actin ring cytoskeleton is an important structure for the formation of sealing zone as an attachment site of osteoclast and bone surface. The pre-osteoclast cells were fused and formed a giant multinucleated cells, which later form a podosome structures and initiate the formation of dense belt-like actin ring. To measure the actin ring cytoskeletal formation, the raw cell were seeded with rankle in the absence and presence of glycocagon A. And then the rhodamin pallidin staining was performed on the day 5 and the image was observed under microscope. The reddish staining of rhodamin pallidin showed that actin ring was clearly formed in rankle-induced raw cells, meanwhile actin ring formation was gradually inhibited with the treatment of glycocagon A. We also measured the expression of integrin alpha, P, and beta 3 protein. This integrin protein localized in the sealing zone and expressed by osteoclast cells. This protein involved in the interaction of osteoclast and bone matrix component. The Western blot analysis demonstrated that in the induction of both integrin proteins was highly enhanced by uncle. However, in contrast, treatment with glycocagon, especially in the concentration of more than 10 micromolar, showed the inhibition of both integrin proteins. This result indicates that glycocagon may fail to form sealing zone required for bone resorption. The expression of integrin protein activates the transcription factor. The CSRC is a tyrosine protein kinase that regulates actin dynamics and organization of protein. This transcription factor generates the phosphorylation of PYK2 and activation of CCBL gene. The complex of CSRC and PYK is required for the normal activity of osteoclast, especially for osteoclast motility. Western blot analysis showed that glycocagon effectively abrogated the induction of CSRC, PYK2, and CCBL increased by the exposure of rankle, indicate that glycocagon A may prevent osteoclast motility. We also measured the expression of vinculin protein, which has played a role in stabilizing actin skeletal formation. Western blot analysis also displayed that treatment with glycocagon A significantly regulated the induction of vinculin dose dependently. The previously mentioned signaling pathway also activates the AKT, which is a signal transduction pathway that promotes cell growth and survival in response to extracellular signal. In this study, the total AKT and the phosphorylated AKT were measured. From the result, we can see that glycocagon A did not affect the total AKT expression. However, the phosphorylated AKT was indeed inhibited, indicating that glycocagon A effectively inhibited osteoclast survival. From this finding, we can conclude that during osteoclastogenesis and bone resorption activity, the interaction between rankle and its receptor ring activate the integrin proteins, which later generates its downstream pathway. The signaling pathway activates the AKT and also vinculin protein, which are responsible for the cell survival as well as the actin organization and osteoclast motility. And then this signaling pathway and the protein is important for the osteoclast differentiation and the activity of osteoclast in bone degradation. However, this mechanism was abrogated in the presence of glycocagon A. Therefore, glycocagon A inhibits bone resorption and actin ring formation by disturbing alpha P beta 3 integrin CSRC, and PYK to pathway. And it demonstrates the potential of lycoplasmin A to be developed as a therapeutic agent for osteoporosis. That's the end of my presentation. Thank you.
Thank you very much for Ibu Lucia Dwi Antika for a very nice presentation regarding about lycoplasmin A as an anti-osteocastogenic activity. Okay, before we going to the next speakers, maybe for your information, if you want to move to the room for participants, except the speaker, you can check the co-host in this room. Thank you. Okay, let's go to the
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity. My name is Waral Faramaida and I'm from School of Pharmacy FPD. I'm lecturer from Faculty of Pharmacy Universitas Jenderal Permadian. This is opportunity I will present my research, result of research, and the other is optimization, extraction, and isolation of sinusoidin from profesional status. In Indonesian name is Berkepribadian. Okay, next slide. The introduction. Profesional status head family Lamiaceae is widely distributed in Africa, Southeastern Asia. The group grows in temperate and tropical areas such as India, Malaysia, China, and Australia. Malaysia and Indonesia have a tropical climate with high temperature and rainfall all year, which have enabled the plant to flourish extensively. There are three varieties of orthosipora status that grow in Indonesia, namely white, purple, and purple pallid. Some of the active secondary metabolites of orthosipora status are sinencetin, eupatorin, rosmarinate acid, and dancetin. Sinencetin is included in the class of flavonoid compounds, and if they are classified more specifically are polymethoxy compounds produced by secretory tissue and stored inside oil gland in plants. Flavon polymethoxy compounds have several pharmacological activities and are part of the plant chemical defense mechanism. Based on the potential and pharmacological activity of sinencetin, efforts are needed to optimize the extraction and isolation of sinencetin from orthosipora status. Objective is my study. This study results are expected to provide information about the composition of food drugs of orthosipora status and usable solvent to isolate sinencetin in acetone extract of orthosipora status tips. This is the orthosipora status plants. The picture A is purple variety, in picture B white purple variety, in picture C flower of purple variety, and picture D flower of white purple variety. The methods of our research, optimization extraction. On the first day, the two varieties of leaves and stem of orthosipora status weigh 1 gram each and add 5 milliliter of acetone. Then let stand for 24 hours. The second day of the TLC filtrate profile was observed. And then the residue was added again with 2.5 milliliter of acetone. On the third day, the TLC filtrate profile was observed. And then the residue was added again with 2 2.5 milliliter of acetone. Therefore, was observed profile of TLC tetrate 3. The mobile phase used in TLC observation is chloroform and ethyl acetate. So the isolation, the step, the extraction of plant material, and the second isolation continue to vacuum liquid chromatography, and purification and identification with TLC preparative and isolate identification. So result in discussion, the result of the optimization of sinenstatin, the extraction with monitoring parameters of the TLC profile, using the result of the optimization of sinenstatin extraction with monitoring parameter of the TLC profile showed that on the fourth day, fluorescence with fourth days, fluorescence from sinenstatin was fading on acetate extract of leaves and stem of 2 varieties of Orthosiphon aristatus. The dry matter ratio used with solvent to maximize sinenstatin withdrawal is 1 gram compared 15 milliliter. Monitoring the TLC profile, the fluorescence of the sinenstatin purple variety is bigger than the white purple variety. This shows that the purple variety level is higher than that of the white purple variety. Earlier research or previous research report that the purple variety of sinenstatin level were higher than white variety. So this is the step, the first step in isolation. In the separation, we take using vacuum liquid chromatography, using a mobile phase of chloroform compared to acetates, which anisocratic system. We have H subtraction, where obtained subtraction 1 to 5 contains the sinenstatin compounds with retention factor 0.45. But what is continued to the separation stage is subtraction 5, because there is not too much chlorophyll, so it is easier to isolate. The second step of isolation, further separation of SF5 using preparative thin layer chromatography with the mobile phase of chloroform methyl acetates. We have 2 bands. The second band is spared by scraping and then dissolved in a mobile phase to filter, filter, and identify the filtrates. And the band 2 filtrate isolate was identified. Band 2 filtrate isolate was identified using TLC and sinenstatin standards. There also saw the same RF between isolate and standards. And our conclusion, the optimal composition of crude drugs and solvent used in the extraction process is essential to maintain the quality of the quantity and quantity of the extract produced. The ratio of crude drugs and solvent for extracting Orthosiphon aristatus, which is 1 gram, compared to 15 milliliter from 100 gram of crude drugs. Sinenstatin was successfully separated from the acetone extract of the purple variety of Orthosiphon aristatus by fractionation using vacuum liquid chromatography and further separation using preparative thin layer chromatography. Okay, this is my last presentation. Thank you for the kind attention. Thank you.
Thank you very much, Bapak Fahrul Faramayuda, for the nice presentation regarding about the optimization, extraction, and isolation of sinenstatin for Orthosiphon aristatus. Okay, before we go into the next speaker, maybe you can chat write in the chat room to ask to the speakers. And okay, I will call to the third speaker, Muhammad Yanis Musja, is there in here? Bapak or the representative of Bapak Yanis. If there is nothing, maybe we going to the fourth speaker, Ibu Sylvia G. Alekatompesi. She will deliver the presentation with the entitled Inner Potential of Bacteria Endophytic from Dayak Onion Plants, Elaterina sp. as an antibacterial Staphylococcus aureus. Please, for Ibu Sylvia, the time is yours. Thank you.
Good afternoon, ladies and gentlemen, participants of the 6th International Symposium of Applied Chemistry. Thank you, moderator, for giving me the opportunity to present my research with the title Inner Potential of Bacteria Endophytic from Dayak Onion Plant as an Bacterial Staphylococcus aureus. Which is the background of this research? Indonesia is a mega biodiversity country that is in medicinal plant and has the potential to be developed into a source of renewable medicinal raw materials. Currently, the development of medicine is back to the nature. Treatment using traditional medicine is safer and does not pose side effects like chemical drug. One type of medicinal plant that is being developed is the Dayak onions, and the specific characteristic of the Dayak plants are rectangles, single leaf, where the leaves are opposite, like green leaf color, separate leaf tip, upper and lower leaves, surface smooth, the repetition of parallel leaves, smooth leaf edge, ribbon-shaped leaves, and Dayak onions contain phytochemical compounds, namely alkaloid, glycoside, flavonoid, phenolic, steroid, and tannin, which have antimicrobial effect and are a source medical raw material, so that they have the potential to be developed as modern medicinal plant in human life. Why the inner potential of Dayak endophytic bacteria? Because the potential of endophytic bacteria contains many compounds that are useful for human life and have high economic value. This potential becomes an industrial capital because of the extent of new raw materials and does not depend on certain medicinal raw materials. This endophytic bacteria technology-wise the inner potential of Dayak onions. Currently, in the face of the COVID outbreak, we are starting to find solutions to overcome this outbreak by using pathogenic bacteria. It's hoped that this result can be used for obtain source the new medicinal raw material that are used for the wider community. Research proposed to obtain endophytic bacteria isolate of diet plants and to determine endophytic bacteria produce secondary metabolites that have antibacterial activity against the Staphylococcus. As research method, we starting from the determination of diet onion plants, and then sterilized tool and media and others, and isolation, purification of endophytic bacteria from diet onion plants, and then regeneration, the test bacteria to be used. Furthermore, screening for endophytic bacteria that have antibacterial potential against Staphylococcus. Then test the antibacterial activity of the diffusion method. And antibacterial test of ethyl acetate extract of the diet endophytic bacteria isolate, and then TLC test, bioautography, and microscopic morphological identification with gram stain, and molecular identification of endophytic bacteria. Result and discussion. The result of plant determination, diet onions from the irradiated with the species named Eleutrin sp. At the time of the determination, the flower of the diet onion were not growing, so the result, the determination were known. Even though this plant is known from Kalimantan, we need to confirm that the plants isolate are diet onion plants. Result of the isolate of endophytic bacteria from diet onions, namely the tubers, there are 11 isolate, and from the stem there are 4 isolate, and from the leaves there are 3 isolate, and from the root there is 1 isolate. Based on the research that has been done, it was found that the number of endophytic bacteria isolate of diet on the tuber was more than in the other parts of the plant. This was influenced by availability of nutrient in the tuber, which was higher than in the other part of the plant, so that there was a large number of bacteria in the tuber. And the result, the purification carried out, the total number of endophytic isolate of diet was 19 isolate, and following the morphology of a single colony of endophytic bacteria. Result of antibacterial screening in endophytic bacteria against Staphylococcus. BDU7 isolate has a large inhibitory zone area compared to the other isolate. They show the presence of metabolic compounds produced by BDU7 endophytic bacteria in inhibiting Staphylococcus pathogenic bacteria. And BDU7 bacteria are from bulbs. Advanced screening for antibacterial activity are continued on the supernatant and pellet using diffusion method, where the results were seen from the clear zone forms on the BDU7 endophytic bacteria paper disk. Of the 5 bacterial isolate, BDU7 has a high potential compared to other, as it indicated by the clear zone form. And antibacterial test result of the extract BDU7. The inhibition zone forms depend on amount of antibacterial active compound contain the extract of BDU7 antibacterial. The result of TLC tests are method to identify antibacterial compound produced by endophytic bacteria BDU7. There are three spot and bioautography tests show BDU7 produce antibacterial compound that they are able to inhibit Staphylococcus pathogenic bacteria. And the result of BDU7 endophytic bacteria staining show that the bacteria belong to the gram-negative and bacil-shaped bacteria. You can see and this reference. And next, the result of molecular identification of BDU7 isolate. It indicates that the bacteria were Pseudomonas aeruginosa, and this phylogeometry based on 16 sRNA gene sequence from BDU7 isolate. And conclusion, 19 isolate of endophytic bacteria from diet anions. The most endophytic bacteria isolate were from tuberous. There were 11 isolates. There was 7 isolates capable of inhibiting inhibiting the Staphylococcus bacteria. There were 5 isolates to produce an inhibiting zone, one of which was the strongest, BDU7. The result of TLC and autobiography show BDU7 produce antibacterial compounds, and identification result indicate that the bacterium is Pseudomonas in the form of bacille and is a gram-negative bacteria. We have recommendation the need to further test on the other 4 isolate and identification of antibacterial compound against endophytic bacteria isolate, so that they can be applied in the health sector. Thank you. That is my presentation. Thank you for your attention.
Thank you very much for Ibu Sylvia for nice presentation. We have 19 minutes for the question and answer. Okay, maybe we have a question first from Bapak Gunawan Pasaribu to Bapak Rauf. The question is, how differences in phenanthropin yield if we compare between using acetone and other solvents such as ethanol solvent? Thank you. Maybe Bapak Rauf, could you directly answer the question from Bapak Gunawan Pasaribu?
Thank you, Mr. Moderator. Thank you for the good. I think this is good question for Bapak Gunawan. So far, I have not found data on the yield of the isolation of phenanthropin, which extraction use ethanol as solvents. But if there is information about it, we can share the information. However, if you look at the polarity characteristic of phenanthropin, which is polymethoxyflavone group, and phenanthropin is more attracted to or more semi-polar to non-polar solvent. And the previous research that I conducted also resulted in data that the phenanthropin content in the extract in the acetone extract was greater than that of the ethanol extract. And the other studies have also mentioned that the combination extraction using acetone and water attracts more phenanthropin. So I think acetone is good at pulling phenanthropin from the photosipon stimulus extract. I think this is my question, Pak Gunawan and Ibu.
Thank you for the answer. Maybe any comment from Bapak Gunawan? Bapak Gunawan, is it clear, Bapak, the explanation of Bapak Rauf about the compare between acetone and other solvent, yeah? Yeah. Can you unmute the speaker, please, Bapak Gunawan?
Thank you, Pak.
Okay, thank you, Pak Rauf, for the answer. Maybe another question for the. Maybe I have some question for Mbak Lucia. I see that you use glycochalcone A as active compound for anti-osteoclastogenic activities. Could you explain for us why you choose glycochalcone A? Is it your isolation or synthesis, or maybe another source from glycochalcone A? Thank you. Halo, Bu Lucia, mohon maaf, nggak ada suaranya ya. Sorry for Bu Lucia. Could you hello? We cannot hear your voice, please. Okay, maybe for your answer you can write in here in the chat. Yeah, yeah. Thank you. Yeah, sorry, your voice now I can hear in this presentation. Sorry. Okay. Maybe another question from other participants. You can raise hand. You can directly ask to the speaker. Very interesting from Ibu Sofia. She's talking about the Dayak endophytic bacteria, right? Why you choose Dayak onion plant this as antibacterial staphylococcus? Is it different between Dayak onion with an original onion in this study? Thank you. Maybe Ibu Sofia. Thank you.
Okay, thank you for the question. I can answer the question. Why we choose the Dayak onion? Because we want to know the potential from endophytic from Dayak onions, and the potential in endophytic microbe can be explored. We can know about especially antibacteria from the onions, but from onions, others onions, maybe we can onions. That is.
Okay. So you choose Dayak onion, yeah. Maybe you original from Kalimantan, right? Yeah, Ibu Sylvia.
Yes.
Okay. Very interesting study that you choose Dayak onion as antibacterial. Thank you for the answer. Okay. Maybe we have 10 minutes more. Maybe from participants, you can chat in. Oh, okay. We have also a question from Bapak Ahmad Potoni to Ibu Sylvia. He asked the endophytic symbiont, such as endophytic bacteria of P. aeruginosa in your study has antibacterial activity. In other study, it also has antioxidant activity and produce growth hormone, indole acetic acid, and this contribute to its hostspan. Please tell me, why do you research the antibacterial activity? Thank you. So the question is, why do you research the antibacterial activity in your study? Thank you. Please your time.
Okay. Thank you, Mr. Ahmad Fatoni. My answer is inner potential of endophytic bacteria is very much and beneficial for human. Actually, I do more research in field of potential microbe for biological fertilizer. Yes, I can get the bacteria potential like your answer question to me. The bacteria potential produce growth hormone IAA, and again biofertilizer. But in this case, I want to know another potential from endophytic bacteria. Another inner potential, one of which to produce antibacteria, and the result we try with several pathogenic bacteria like E. coli, Bacillus, Pseudomonas, and Staphylococcus. But BDO7 bacteria endophytic is unique, is being able to inhibit pathogenic bacteria, especially, and the bacteria of Dayak onions is a lot of potential. In our research, we must go to the research to antioxidant from Dayak onions. From Dayak onions, we have result from antioxidant, other isolate from Dayak onions have antioxidant too, and this is a challenge for research in Indonesia. And in this research, Professor Simanjuntak helped our team a lot to continue to study the potential that exists in the endophytic microbe like this.
Okay, thank you, Ibu Sylvia. Any comments from Dr. Ahmad Fatwani?
Thank you, Mustafa. Thank you, Bu Sylvia, for your answer.
Okay, thank you very much for the question and the answer from Ibu Sylvia. Okay, Ibu Sylvia have got the answer, the question from for me. He didn't isolate the compounds by himself. They use commercially available liposarcone A. Since possibility of low quantity of targeted compounds, so doesn't isolated by itself. They focus on mechanism of work from liposarcone A. Using commercially available ones is better since it's purer. Why he chose liposarcone A? Because from previous study that reported that liposarcone A possesses various pharmacological activity. Then he wanted to examine the possible beneficial effect of this compound on degenerative related disease, in this case osteoporosis, which the prevalence is keep increasing these days. Okay, thank you very much, Ibu Lucia, for the explanation. Okay, is there any question? Sorry, Ibu Carla also asked to Ibu Lucia. She wants to know, could you share where did you run Western blot analysis of the study? I am interested to do sort of essay. Thank you. Ibu Lucia, maybe test your voice. Sorry, we can hear your voice. Maybe you can write the answer in the chat room, please. Okay. Ladies and gentlemen, finally we come to the end of the presentation. We would like to say thank you again for all the presenters for the informative and interesting talk, and to audience for your active participation. So we can give big applause to the all speakers. Hopefully this presentation will be beneficial for everybody. For your information, for all participants, you have time about 15 minutes for break, and please come to join in the parallel session 2 will be moderated by Dr. Abdi in the 3:15. Thank you very much for your time. Good afternoon. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Terima kasih Bapak Ibu sekalian. Ibu Lucia boleh sambil mencatat jawaban di sini, atau nanti bisa langsung ya ke Ibu Lucia. Thank you.
Okay, good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh. Sorry, a little bit late because we have some trouble here. Thank you very much for staying tuned in our parallel session for this afternoon. So in this afternoon, we still have 6 more presenters who will give the presentation to us. So before we start our program today, it would be much better if I remind you back around about the rule in this parallel session, especially for Q&A. If you have more questions to the speaker, you just chat directly into the chat room, your name _ institution, and then to the presenter. So for the first speaker, here we have Ade Chandra Irwansyah. He's going to talk about his study entitled Study on Antioxidant Activities, Acceptability and Self-Life Prediction of Ciplukan Cialis Angulata Juice Drinks. So please welcome.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you, moderator, for the opportunity. On behalf of the day, let me introduce myself. My name is Ade Chandra Irwansyah. I am a researcher at the Research Center for Applied Technology, Indonesian Institute of Science. For opportunity in this conference, we would like to present our research with a study of antioxidant activity, acceptability, and self-life prediction of Ciplukan juice drink. Next. For introduction, the plant of Cialis angulata or Ciplukan have a functional properties that are used as alternative to traditional medicine, such as anti-hyperglycemic, antibacterial, immunomodulator, and antioxidant. This plant contains some antioxidant compounds such as quercetin, vitamin C, isolate, and organic acid. In the food sector, antioxidant in the low concentration compared to oxidative substrate significantly reduce or prevent the adverse effect of oxidative stress on the normal physiological function of human. In the content of alkaloid, polyphenol, flavonoid in the Ciplukan leaf and fruit extract test in the anti-hyperglycemia activity in the pancreatic beta cell. Our previous study regarding the characteristics of each part of the Ciplukan plant showed the highest antioxidant activity in leaf, root, and bud part. Utilizing the antioxidant content in the Ciplukan carry out the development of functional food-based product, namely Ciplukan juice drink. Therefore, the botany of this Ciplukan plant has been carried out at Herbarium Bogoriensis Research Center for Biology, Indonesian Institute of Science. And for the citric acid, stevia and CNG were obtained at Stealina Chemical Store in Bogor, while honey is bought at the Sivinoviva Bogor. For preparation sample, the fresh Ciplukan fruit and bud are soaked, weighed, then washed with the running water. The extraction process using a water solvent with a ratio 1:2 and 1:20. Then filtered, mixing juice extract and bud extract of Ciplukan with a variety ratio. After that, the extract was pasteurized at a temperature 70 degrees Celsius for 15 minutes, then packaged in a sterile bottle. For experiment design, the result was used a factorial completely randomized design with 2 factors.
The first factor is the ratio of fruit juice extract and the bud extract. And the second is the level of stabilization. The parameter test in this step were sensory quality or hedonic test and the antioxidant activity. Analysis of total phenol, total flavonoid content, and alkaloid content was carried out on the two base samples based on the sensory test result and antioxidant activity. The base formula were then analyzed for estimating the cell life by Arrhenius method for 28 days, which every 7 days observing parameter of vitamin C level, pH, and total microbial count. Result and discussion. For sensory parameter, in this figure we can see that the extract ratio between fruit and bud extract of Ciplucan 20N1 or IA4 had the highest average value of reference test. The more fruit Ciplucan extract in juice are added, the tendency for panelists to prevent test increases. Despite in Ciplucan feed, there is a sugar content that can add a sweet taste in the drink. Meanwhile, in the bud, alkaloid, tannin, and saponic compound have a bitter taste characteristic. The fruit and bud extract with ratio 10:1 or A2 had the highest average value of flavor attribute. It might be the aroma associated with the volatile compound in the food. The more volatile compound and the form of aroma will be subtle. The ratio of juice and bud extract of Ciplucan and stabilizer concentration significantly affected of Ciplucan of color attribute. We can see that the extract of fruit and bud extract with a ratio 15:1 and 20:1 with a stabilizer concentration of 0.1%, namely A3B1 and A4B1 had the highest preference for the color attribute of the Ciplucan juice drink. The juice extract ratio and the Ciplucan bud and stabilizer concentration significantly affect the viscosity attribute. The treatment with ratio 15:1 with the stabilizer concentration of 0.1% had the highest preference value on the Ciplucan bud extract color attribute. According to Kumalasari, report that the higher of the stabilizer concentration, the higher the viscosity of juice. For antioxidant activity parameter, in figures 2 show that the ratio of fruit and bud extract of Ciplucan, stabilizer concentration, and their interaction significantly affect antioxidant activity. The best treatment of antioxidant activity demonstrated by a sample with fruit and bud extract ratio 5:1 and the CMC concentration of 0.3% is the 73.45 microgram per ml. For result of total phenolic content, flavonoid and alkaloid, the base sample is chosen based on the highest number of organoleptic test results from all the attribute assessed and antioxidant activity. Based on this, two samples of the best Ciplucan juice drink were obtained, namely A3B1 and A4B1. The highest total phenolic and flavonoid content were obtained from sample with a juice fruit extract ratio and bud extract 15:1 and CMC concentration of 0.1% with a total phenolic content 18.74 milligram gallic acid equivalent per liter and total flavonoid content 0.25 milligram quercetin equivalent per liter. The highest level of alkaloid content with the extract ratio 20:1 and CMC concentration of 0.1% with amount 6.61%. The shelf life of Ciplukan Jus 3. In this slide, we can see that figure 3 shows that during storage at a room temperature, there is a decrease in vitamin C level. The regression equation for decrease of vitamin C level in the Ciplukan functional tree is Y equal minus 0.425 minus 0.0229x plus 6.09 with R squared 0.953, which means a constant decrease in quality 0.0229. And figure 5 shows that during room temperature storage, an increase in the total plate count values cause a decrease in the product quality. The regression equation for the TPC value of the Ciplukan functional tree is Y equal to 55.571x plus 824 with R squared 0.99, which means a constant decrease in the quality 55.571. With respect to the shelf life of Ciplukan juice, the table shows that the Arrhenius equation with its length K equal to minus 824 minus 555.571 1/t, and the activation energy of 110.363 calorie per mole. So the value of the constant rate of decline in the quality of the TPC parameter obtained is 5.5829 per day. By using the equation, so we can see that the storable of Ciplukan juice drink based on the TPC value at room temperature is 9 months. Last but not least, with respect to Ciplukan juice drink analysis, the extract ratio of juice and bud extract influence the flavor and aroma attribute in the sensory evaluation. The interaction between ratio juice and bud extract and stabilizer concentration influence the antioxidant activity, color attribute, and viscosity of Ciplukan juice drink. The best Ciplukan juice drink sample based on sensory evaluation and antioxidant activity were A3B1 and A4B1. The highest total phenolic and flavonoid content of Ciplukan juice drink were A3B1, meanwhile the highest alkaloid content was A4B1. The shelf life of Ciplukan juice drink is 9 months. For acknowledge our deepest gratitude is sent to Insinas Research Grant 2020 from the Ministry of Research Technology BRI, Republic of Indonesia, and Indonesian Institute of Science ELSA for financial and technical support. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much for the presentation. So now we move to our second presenter today. Here we have Muharmi. She's going to talk about anti-hyperlipidemia activity of ethanol extract, distembak of Flacotiarucam on propylthiouracil-induced albinolet Rotus novaticus Swiss strain. Please welcome to our next speaker.
Assalamualaikum warahmatullahi wabarakatuh. I want to presentation my research anti-hyperlipidemia activity of ethanol extract, the symbol of Flacotiarucam on albinoret Rotus novaticus Swiss strain in propylthiouracil-induced. Tim riset Muharmi, Julinar, Heni Yohini, Fitria, Rimanati, from Department of Chemistry, and Department of Pharmacy, Faculty of Mathematics and Natural Science, University of Sriwijaya, South Sumatra, Indonesia. In previous research, three compounds, predelene, terroid glycoside, and polyacryloside have been isolated. Ethanol extract, lacutirocam, also show in vitro hypertension activity and in vitro anti-cholesterol activity. In this research, I want to report in vivo anti-hyperlipidemia activity, testing of lacutirocam, and then was determined effective dose 50. Propose the research determine in vivo anti-hyperlipidemia properties testing of lacutirocam using male albinorate blister strain using the enzymatic colorimetric method. And then determine effective dose anti-hyperlipidemia activity ethanol extract testing of lacutirocam. Methodology: animal test to induce by profile thiouracil 1% and in condition animal hyperlipidemia. And then to divide it 6 group, normal, control negative, control positive, dose 1, dose 2, and dose 3. And then to treatment with ethanol extract, zestrin of lacutirocam. After 44 days, cholesterol total, triglyceride, LDL, and HDL value was determined, and then effective dose 50. Result and discussion. Total cholesterol value. The total cholesterol after acclimatization is in the normal range. After induction, profile thiouracil, total cholesterol increasing level above normal. After treatment, show the decrease in total cholesterol level. The highest decrease in giving by detriment of dose 3 manual simvastatin F positive control, so decrease in total cholesterol level, which is the same as treatment dose 400 mg per kilogram body weight. The statistical analysis show positive control, group 1, 2, and 3 show significant difference, and no significant difference between the treatment dose of 400 mg per kilogram body weight with positive control. Based on regression, linear equation obtain effective dose. Triglyceride. The triglyceride value of normal is 25 to 145 mg per kl. The rate induced by PTU show significant increasing in triglyceride level, but in the normal group show not significant increasing in triglyceride level. The given ethanol extract at dose 100, 200, and 400 and positive control simvastatin show significant decrease in triglyceride level compared to before treatment. The triglyceride level between group before and after treatment show positive control. Group 1, Group 2, and 3 show significant difference, and treatment Group 1, 2, and 3 could reduce triglyceride level significantly. Effective dose triglyceride was determined based on the linear regression between the dose of ethanol extract and the percent decreased triglyceride level. Measurement LDL. After inducing, the average increase in LDL level. The decrease of LDL level after being treatment show significant difference compared before treatment. The decrease in LDL level for positive control also show significant difference. Significant decrease. The LDL level between group before and after treatment show positive control. Group 1, 2, and 3 treatment show significant difference. Determination of HDL. The measurement of HDL level after acclimatization show that HDL level in normal range. The giving profile uracil and high-fat food causes HDL level to decrease significantly. The treatment with ethanol extract from shows significant decrease in HDL. The HDL level between group before and after treatment show a positive control. Group positive control 1, 2, and 3 show significant difference. Presented increased HDL was determined based on linear regression equation between extract dose with present increased HDL. After the treatment, the positive control group and the treatment group dose 1, 2, and 3 show significant different weight compared with before treatment, whereas for the normal group and negative control show increased body weight, significant difference. Conclusion. Treatment group are showing significant reduction of total cholesterol, triglyceride, and LDL, and significant increment of average HDL compared with the negative control. The ethanol extract from the stomach of placurtiarukam have anti-hyperlipidemic activity that not only can reduce total cholesterol, triglyceride, and LDL level, but also can increase HDL level in moderate with effective dose 3028.09 milligram per kilogram body weight. Acknowledgement, the author are thankful to the respected Republic Indonesia who have funded this research, and thank you also to Department of Chemistry, University of Sriwijaya for providing facility for carrying out the research work. Thank you.
Thank you very much. For the presentation to our speakers, Megawati. Megawati will deliver her research about inhibition activity of alpha-glucosidase enzyme, phenolic content, and toxicity of Mara Macaranga Tanarius leaf. Please welcome.
Everybody, my name is Megawati. Today I want to explain about my research. The title is Anti-Diabetic Activity, Phenolic, and Toxicity of Mara leaf or Macaranga Tanarius. This is my outline. Introduction, research method, result and discussion, and conclusion. The genus Macaranga is useful species of 250 species in tropical forest, where its distribution is in tropical forest of Asia Pacific. Investigation of the potential of Macaranga plant as medicinal plant as cosmetic ingredient is still not optimal. It phytochemical and pharmacological studies of Macaranga plant that have been carried out generally show that there are variations in the content of secondary metabolites such as terpenoids, phenylated flavonoids, and stilbenoids, and some phenolic derivatives. More than 26 species of Macaranga have been studied by phytochemical with more than 190 secondary metabolite compounds already identified, with phenolic compound as the major compound group. One previous polychemical study of Macaranga genus collected from Mekonga forest, where success isolated and identified some phenolic compounds such as macarangin, apigenin glycoside, and scopoletin from Macaranga gigantifolia, campherol, piperenil flavonoid, and scopoletin from Macaranga spida. Apigenin and active compound were isolated from Portulaca as potential antibacterial activity and can be used for drug development as an antibacterial for the treatment of disease associated with the pathogenic bacteria. Phytochemical content of the Macaranga genus needs to be done, especially from Macaranga tanarius or Mara. We evaluated the total phenolic content from methanol extract and hexane ethyl acetate and butanol fraction and antiderivative activities from Macaranga tanarius. It's a research method. The dry leaf 2 kg of Macaranga tanarius was macerated with methanol for 24 hours 3 times. The methanol extract 220 grams were partitioned with N-hexane, ethyl acetate, and butanol. And all fractions were evaluated for their total phenolic content using Pauling-Carteau method, inhibition activity of alpha-uric acid, and toxicity test. This is result. In this study, the Simplicia used was a live which had been pollinated the Mara leaf when the extract using 96% methanol. The extraction method used is the maceration. Maceration is done 3 times by soaking 2 kg of Simplicia powder in maceration bottle. After all the macerated were collect, solvent separation was carried out using vacuum rotary evaporator. The solvent liquid can be separated without any overheating of the sample by lowering the pressure above the liquid and thus lowers the boiling point. After obtaining methanol extract of Mara leaves as much as 220 grams with percent yield 10.33% and the yield extract and fractionation of the hexane fraction get the extract weight 1.522 gram is 7.6% yield. Fraction ethyl acetate get the extract weight 9 gram with the percent yield is 45.22%. Fraction butanol get the extract weight is 5.88 gram and the percent yield is 29.40%. And the last water fraction get the extract weight 3.21 gram and the percent yield is 19.09%. In the total phenolic content of methanol extract was 27.117. It means that each 100 gram dry weight of the methanol extract contained total phenolic that was equivalent to gallic acid addition step of methanol extract with hexane, ethyl acetate, and butanol yield hikes total phenolic content in the ethyl acetate fraction with the total phenolic content was 37.518. Ethyl acetate fraction was further purified using various chromatography techniques. This is the data of inhibition activity of alpha glucoside. The support by the data analysis for biological activity test. The results show the ethyl acetate of Macaranga tanarius leaf has strong activity as antidiabetic with IC50 0.74 ppm compared with quercetin as positive control with IC50 5.60 ppm. In this data, AL fraction, hexane fraction, ethyl acetate fraction, and butanol fraction get IC50 under 25 ppm. With the literature, the extract the fraction, this is active and have inhibition of alpha glucoside. This is data of toxicity test. Toxicity tests are used to determine the toxic effect of a compound that will occur is the short-time exposure period of the administration of single concentration of the compound in test animal. The BSLT method is used because this method is easy, inexpensive, fast, does not require special lab, and the results are reliable. In this table, the only fraction and hexane, which is a toxic compound that has N is IC50 300.99 ppm. Compared to other extract, is not classified as a toxic compound because it has IC50 more than 1000 ppm, which according to the theory that N is declared toxic according to the BSLT method if it has LC50 of the less than 1000 ppm. From the result obtained, it turned out the ethyl acetate fraction has resistant value of alpha glucoside of 0.74, more active and approaching the standard quercetin 5.6 ppm when compared the other extract. In the BSLT test, the ethyl acetate extract didn't show any cyanarrhythmia larva death after adding this extract. Extract show toxic activity in BSLT if the extract could cause the death 50% of test animals as concentration of less than 1000 ppm. The statement above show the ethyl acetate extract is not toxic to arrhythmia because it has LC50 value under 1000 ppm. The ethyl acetate extract in the phenolic content test has higher content. Alpha glucoside is said to consume without showing mortality in this extract when test for toxicity by the BSLT method. This is my conclusion. In the Macaranga tanarius leaf extract from the ethyl acetate extract has the main phenolic compound, which has the potential to inhibit the enzyme alpha glucoside. There is forceful correlation between the ability to inhibit alpha glucoside and the safety of the compound contained in the ethyl acetate extract, which doesn't show its toxicity. So the ethyl acetate extract from Macaranga tanarius or MARA leaf can be declared as candidate for diabetic drugs. Thank you for your attention.
Thank you very much. Now we proceed to our fourth presenter. Now we have Mr. Reinhard Jeming. He's going to talk about his research entitled Bacchia fruit essential oil was tested with Aedes aegypti larvae at various concentration level. So please.
Good afternoon, ladies, gentlemen, and all of the participants. My name is Lira Asaria. Today, I would like to present to you a presentation with the title Activity of Essential Oil of Ujung Atap Leaves Against Aedes aegypti Larva. This research was conducted by Renhat Jemi, Sumiati Simanulang, Nuwa, Sarina, Desi Natalia Koro, and me myself. We are from University of Palangkaraya. So when we talk about dengue hemorrhagic fever, it's a very common disease here in Indonesia, especially at some places, which is in Central Kalimantan itself, especially in Kuala Kapuas. This disease was concluded as an outbreak in 2018. So that many households use a synthetic mosquito repellent to prevent the mosquito from coming, because this disease is caused by Aedes aegypti mosquito. But sadly, the synthetic mosquito repellent has a toxic effect for human being. From that problem, we are searching for any alternatives, which is natural insecticide. Actually, here in Central Kalimantan, the Dayak tribe use Ujung Atap leaves as a mosquito repellent in order to prevent the mosquito from coming. So that we do a literature review about these leaves, and these leaves has so many benefits such as anti-inflammatory, antioxidant, antifungal, antibacterial, and also antimicrobial. From that, there is a possibility that Ujung Atap leaves could be an anti-Aedes aegypti larva. That's why this research was conducted. So this is our research method. We gather the leaf first, the Ujung Atap leaves. We divide it into two, the dry one and then the fresh one. After we divide it into two, we do the distillation. When we did the distillation, the leaves, there are the ratio of the leaves and also the water. The ratio is 1/2. Every 1 kilogram of leaves, we will add 2 kilos of water. After we did the distillations, finally we got the essential oil, so that we can analyze the properties. We can do the larvicide test, and also we can do the DCMS analysis itself. After that, we can have a result and discussion about this research. And this is the result and discussion of our research. As you can see here in the table, there are quite different results on dry and fresh leaves, primarily on the random number, the acid number, and also the ester number, and the specific gravity itself quite different, but the color and the smell is the same. We also compared the dry and fresh leaves of Ujung Atap essential oil with eucalyptus oil. Why? Because they have the same family. They came from the same family. And then we do the larvicide test. And this is the result here in the graphic. As you can see, the blue one represents fresh leaves, and then the red one represents dried leaves. As you can see here, we got the result here in the table that these leaves would effectively kill the Aedes aegypti larva. And And this is our GCMS analysis. As you can see here in the table, the fresh leaves contain 25 compounds, meanwhile the dry leaves contains 23 compounds. There are some compounds that is available in fresh leaves but not in dried leaves. And also there are some compounds that is available in dried leaves but not in fresh leaves. So you can see here there are limonene and also bicycle and so on. And from the literature review that we got, there are 3 antifungal compounds in both dry and fresh leaves, which is alpha-pinene, beta-pinene, and also eucalyptol. Pinene here is believed to be the compound that gives scent on the essential oil. That's why both dry and fresh leaves has the same smell, the same scent. So here we are at the conclusion. So based on the result, the ujung atap essential oil, the dry one produces a lot more essential oil than the wet ones, than the fresh ones. And also there are different compounds in dry and also fresh leaves. The fresh leaves contain 25 compounds, meanwhile the dry one contains 23 compounds. And one compound that is believed could kill Aedes aegypti larva is limonene, which is contained in dry leaves, and also there are other minor changes. Thank you.
Right, thank you very much for this presentation. Now we move to our fifth presenter today. We have Dr. Galuh Widiarti from LIPI. She's going to talk about antioxidant activity of botanical extract from Madanggatal, Himalayan choice leaf. So the floor is yours.
Good afternoon. Thank you to moderator. Now I will present our research with the title Antioxidant Activity of Botanical Extract from Madanggatal, Himalayan choice leaves. I am Galuh Widiarti from Research Center for Chemistry, Indonesian Institute of Sciences. The outline of our presentation consists of introduction, objective of the research, material and methods, results, and conclusion. Introduction of the research care about the background of our research. Everybody knows environment risk of the free radical, and the free radicals can contribute to the aging process of tissue and cause various chronic and degenerative disease in humans, such as cancer and cardiovascular problem. The risk can be reduced by utilizing of radical scavenger compounds known as antioxidants. Indonesian biodiversity sources of bioactive compounds as antioxidant agent, especially Indonesian medicinal plants. One of the interesting plants under study as a potential antioxidant is Madanggatal, Himalayan choice TC, and the plant locally known as Puspa or Celone. From the literature, some bioactive compounds has been isolated from the plants, where kaempferol or ramusep as anti-malarial, anti-cancer, and anti-plasmodial. The other known as antimicrobial and antifungal, and theanine has psychoactive properties for depression, stress, insomnia, sleep disorder, hypertension, and some symptoms of schizophrenia. The purpose of the study is the antioxidant activity of the butanoic extract from madang catalyst. The antioxidant activity of the extract will be studied based of the radical scavenging effect of the stable BPPS radical promoter. The material used of the study were madang catalysts that collected from kebun provinsi Pustitek, and the dread extract and the dread pipes were macerated using methanol. After that, the methanol extract was shown using normal hexane, ethyl acetate, and methanol respectively. And all the solvent used for maceration and purification and percussion were the technical solvents, and some analytical grades from America, including silica gel oxides and sulfuric acid for the analysis, methanol and acetic acid for analysis, DPPS for antioxidant activity analysis, and silica gel for column chromatography to provide the active compounds. And the equipment used for the research were after IRC Matsu practice 21 using KDR to determine the functional groups, and LCMS marginal biospectrometry, electrospray or IS1 system to determine the organics of the result. The method of the research first, the list of madang kata will be conducted using DPPS methods, and the purification of the extract using silica gel column chromatography, looking with normal hexane gradient of ethyl acetate to 100%, and then isolation of the fraction that most active as an antioxidant was conducted using column chromatography and continued by preparative TLC, and the isolate was then identified using TLC, FDIR, and LCMS. The result first, the yield of madang catalyst. From the table, we see that the high-risk extract was methanol extract with the yield around 20.18%, and normal hexane extract 0.06%, ethyl acetate extract 2. All the fraction and the butanol extract, we analyze the antioxidant activity and compare to quercetin as antioxidant standard. From the antioxidant activity research, we see that the fraction B2 has highest antioxidant activity with IC50 value about 24.50 microgram per milliliter, and so the fraction is categorized as very active as an antioxidant. Nevertheless, the antioxidant activity of the fraction less active than quercetin as antioxidant standard. And the fraction B2 that most active as antioxidant, we continue to provide using column chromatography and preparative TLC, and the yield one isolat, and the isolat was identified using IR and LCMS. And from the IR spectra, we see that the IR spectra of the compound has similarity with catechol compound as reference material, where the with number at around 2,246 centimeter, similar with absorption band of catechol at with number 2,363 per centimeter, indicating the presence of hydroxyl groups at with number of 1,608, indicating the presence of the carbon 6 groups similar with abscission and catechol at number at 1,611. The LCMS result showed that the chromatogram illustrated one main peak at representation time of 1.68 minutes and an ice of 290.25 gram per mole was recognized as catechol with MS 290.30 gram per mole. And from the literature, so that catechol and cyanin were main compounds commonly found in TC plants. From our research, we conclude that the first butanol extract was very active as an antioxidant with activity value of 12.55 microgram per liter. The purification and isolation of the extract produce one isolate amount of 15.5 milligrams with high spectrum as flavonoid, and based on IR and NS spectrometry, the main compound of isolate from B2 may be a catechol compound which has some functional groups similarities with RS 290.25 gram per mole. And the isolate also has strong antioxidant activity with activity value of 17.97 microgram per milliliter. So that we conclude that madam batanol is potentially to be developed as a natural antioxidant for preventing human disease. And from the research, the suggestion from the research is identification of the isolate using INR should be carried out in the next research. And we also thank to Mr. Suratman for his help collecting Madam Batal's sample, and Dr. Hatiwi and Mrs. Purwati for their suggestion for our study. Thank you so much for your attention.
Thank you very much for the presentation. We apologize. Just now we have heavy rain here and thunder. We apologize for some trouble. So now we proceed and we go on for our last presenter today. We have here Mrs. Husniati. She's going to talk about anti-cancer activity and Lampung Robusta coffee extract fraction. Please welcome.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon everyone, especially Indonesia time zone. Greetings was also conveyed to all participants who were willing to attend this time meeting. Hopefully it will be of benefit to all of us. Amin. In this occasion, I would like to present our research. The title is Anti-Cancer Activity of Lampung Robusta Coffee Extract Fraction. This paper was written by Husniati et al. And I am Husniati as a senior researcher in Center for Research and Standardization of Industry, Bandar Lampung, Indonesia. In order to start my presentation, I will explain briefly outline of this presentation. The first part is background, literature review, material, method, and result, and the last part will be conclusion. Refer to reference by Fujioka and Shimamoto 2008. Coffee include the second rank of all food commodities that are consumed and traded worldwide. According to data from International Coffee Society, Indonesia fourth in the world as a coffee producer after Colombia. Indonesia is one of the biggest exported country, especially in agricultural product. As we can see this slide, one of the main plantation commodities in Indonesia is coffee. Lampung is the second biggest coffee producer province in Indonesia after South Sumatra. Lampung has a very potential in national coffee production, and Robusta coffee is the greatest amount of coffee than Arabica coffee. Coffee contains various compounds such as coffee metabolic. CGA is important and biological active dietary polyphenol, playing several important and therapeutic roles such as antioxidant activity, hepatoprotective, antivirus, antidiabetic, and antihypertensive. This research focused on the variation of the mobile-based solvent composition in product purification of CGA and coffee bean extract fraction, with that analyze the anti-cancer activity of extracts. The research applied fractionation based on mobile-based polarity. The variation of mobile-based composition is composed from polar to non-polar organic solvent with gradual addition of non-polar solvent. The objective of this variation is to select and separate compounds based on compound polarity from high polar compounds to less polar compounds. This research studies correlation of the solvent to the extraction product characteristic. The different solvent composition used in chromatography process. It's a seven sound. Benefit research provide information about natural compound from Robusta green coffee beans as an alternative therapeutic treatment and as a material from new pharmaceutical product in future research. The main material is organic green Robusta coffee bean from Padang Cermin, Lampung, Indonesia. The coffee beans were determined by Herbarium Bogorians Research Center for Biology, LIPI Bogor as Caffea carnivora Rohner. Our research are start from extraction, following by partition, fractionation, chromatography column, CGA determination, and anti-cancer test. So this slide shows the process of extraction. Green bean coffee was extracted with methanol. The extract was concentrated with the rotary vacuum evaporator to obtain the methanol extract using TGA and caffeine caffeine standards. Detection of TGA. qualitative analysis using TLC and quantitative analysis using qualitative analysis using TLC and quantitative analysis using PLC method. Test for anti-cancer activity using MCF7 breast cancer cell. We obtain MCF7 cell line from LIPI. Presence of CGA and anti-cancer activity in its sample fraction is present in table 1. This table also mention about the color of the fraction as determined visually. For better visualization, the complete picture of all fractions are displayed as figure 1. The determination of CGA absence was conducted by visual determination on the spot by TLC analysis compared to CGA standard solution. The picture about mentioned TLC analysis on CGA standard solution, the TLC analysis observed that there was no CGA substance in sample G. Its fraction was tested again, MCF7 breast cancer cell line, in order to study its anti-cancer activity. The G fraction was beyond the sample cytotoxicity again MCF7 cell line, although it didn't show any presence of CGA based on the TLC analysis. Our sample particular fraction A and B which contain CGA, however, do not indicate any anti-cancer characteristic. There are some possibility beyond this negative result. First, anti-cancer activity of CGA is dose and time dependent. The concentration of CGA in both fraction A and B were too low to show any anti-cancer effect. Second, there are compounds other than CGA in both fraction A and B with anti-cancer effect. In addition to CGA, it is suggested that several compounds from coffee such as caffeine, ferulic acid, and caveol, which have anti-cancer characteristics, as displayed in either in vitro or in vivo studies. The G fraction in this study needs to be investigated in the future research to identify the potential anti-cancer compound. This is conclusion for this research. Separation of coffee extract using a polyamide column from chromatography with water methanol solvent produce fraction with different compound composition. TLC analysis shown that fraction A and B contain CGA substance. Only fraction G has anti-cancer activity against MCF7 breast cancer cell line, even though it doesn't contain CGA. Finally, I was thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much for the presentation. So now we go on to the discussion session. Here we have I think question to Reinhard Jimmy is already answered, but some still not yet. So first question from Sofa Fajria to Ade Chandra. Why the optimum shelf life of your Ciplukan drinks only 6 months? Can we extend the shelf life? Do you edit the preservative agent in your research? Thank you. Please right away to answer this question. Mr. Ade Chandra.
Okay, thank you, moderator. Thank you for Miss Sofa Fajria. I think it's correction, not 9 months, but not 6 months, 9 months. Okay, for the shelf life of juice drink. The 9 months is based on the maximum limitation of TPC value in standard national Indonesia. There is 10,000 colony per mil. If we pass the value of maximum limitation, it means that the juice was broke, or the juice was broke. Sorry. For the preservative, we just add the citric acid for the food additive. I think it's my answer. Thank you. Okay.
It's clear? Okay. Is it clear, Dr. Stofa?
Hello? Is it clear?
Clear, Dr. Thank you. Yes.
Okay.
So we move to the next question from Melati to Mr. Reinhardt. Will the efficacy of brachiaprostesen essential will be the same of Aedes aegypti mosquito? Not in larva form, so it can be applied as anti-repellent cream or lotion, etc. Is there any further plan for efficacy study on mosquitoes? Please right away, Mr. Reinhardt, Jamie, to answer this question.
Okay, thank you, thank you. Mana dia tadi? Mrs.
Question from Mrs. Melati.
Okay.
Mrs. Melati.
Okay, thank you, moderator. Okay. Thank you, Mr. Melati from LIPI. Very impression question. Essential oil from the tip of the roots. The root is used as inhibition. Method 1 gram the from measured oil, and now are the plant mixture replaces made from essential oil from and root cream. Thank you, moderator. Okay, thank you, moderator.
Miss Melati's question.
Melati, ini kan?
Yeah.
Okay, thank you for the answer.
Okay.
Okay, next from one question from Mr. Reinhardt Jamie to Elvita. The difference, sorry, this. I'm sorry, I'm mistake. Sorry, from Mr. Gunawan Pasaribu to Husniati. Do you have any comparison bioactivity data with roasted coffee?
Yes, okay.
Okay, we can hear you clearly.
Thank you for Mr. Gunawan Pasaribu. We have compared with roasted coffee for antioxidant activity. There is a decrease as much as 70 until 90%, maybe. The answer. Thank you.
How is it, Mr. Gunawan Pasaribu?
Okay.
The response.
About the anti-cancer activity, Bu?
Maybe it's the same result because we interest for CGA activity from antioxidant, and then we found from extracts. Okay. Sorry. Based on a number of extracts with the highest antioxidant activity showed the best anti-cancer activity. This is the point, my point for our research.
Okay, thank you. Thank you.
All right. So we move to the next question from Ahmad Fatoni to Mr. Adi Chandra. Ciprucan, based on your study, has antioxidant activity due to its phenolic content in the extract. Can you tell me, is there any correlation of the extract content, such as phenol and alkaloid, to contribute for itself or life or not?
Okay, I think there is a correlation of the total phenol or alkaloid to self life. As we know that the total phenolic inside some phenolic content, have ability as antimicrobial, and also with alkaloid compound. For this ciplucan, they have a microbial. I think it's yeah.
How is the answer, Mr. Ahmad Patodi?
Oh yeah, sorry. Thank you, Mr.
Okay. Thank you.
You're welcome.
All right. Next question from Dr. Sofa Fajria to Ibu Husniati. In your results show that fraction G, which has highest anti-cancer activity but did not contain CGA. Could you explain us about this, Ibu Husniati?
Yes, thank you for question, Ms. Sofa. For the answer to this G fraction, I think no, they fraction G is not CGA fraction, not same CGA fraction, and then we this time we cannot predict this anti-cancer compound. Maybe next we compound which give the anti-cancer reduction effect are still debatable, maybe because we need it to next research. Thank you.
Thank you.
Any response, Dr. Sofa?
Yeah, are you doing our CGA anti-cancer activity?
Yes.
But how about the result? Is it good result?
No, breast cancer breast cancer. The CGA is not activity anti-cancer breast cancer.
But from maybe from the literature, an activity, right? Yeah.
Maybe to presentation I mentioned about our research. We didn't find anti-cancer breast cancer. Maybe this one. The CGA is have a minimal dose, maybe too low to show cancer effect. And then yes, maybe our conclusion. G fraction have CGA fraction didn't have anti-cancer focus to breast cancer activity. Maybe they have low dose to inhibit this cancer.
How is it, Dr. Sofa?
Thank you. Maybe later we can discuss again.
Yeah, thank you so much, Ibu Husnaini, for your response. Now we move to the next question from Prof. Andrea Agusta, asking question to Ibu Muharni. Your research show your sample posing anti-hyperlipidemia activity with certain ICVT value. So this is potentially enough to use for human. Please respond right away. Bu Muharni.
Thank you, moderator, and Prof. Andrea Agusta. This flow corticosteroid is one of traditional medicine has been used for anti-hypertension. Based on this research, we to scientific the use of plaque artery come to hypertension. Based on AC50, we have obtained less AC50. And then we conclude that plaque artery is potential to use of treatment of hypertension. Thank you, moderator.
How is it, Prof. Andrea Agusta? Do you have any response? Is the answer?
Thank you for the answer to the question. Yeah, maybe this is very interesting. Yeah, maybe I would like to try to cure some hypertension. Maybe you can recommend.
Yes, hypertension based on trigger of hypertension is one of level anti-cholesterol activity. Anti-cholesterol. Well, cholesterol activity is high in blood in hypertension. To trigger hypertension as one of trigger hypertension based on value of cholesterol, high cholesterol in the blood.
Okay. Thank you very much, Ibu Muharni.
Okay, we move to the next question from Lucia, Ibu Husniati. Have you checked the effect of your extract in normal cell line for comparison? Since it's important to determine the toxicity of any sample used into the normal cell to make sure that your compound is not harmful. Please right away, Ms. Husniati, respond this question.
I will share screen. This is a test for anti-cancer activity. Anti-cancer activity for a positive. Saya lagi seminar. We use solvent control, positive cell control, was prepared by transferring and incubating cancer cell in RPMI, and then I saw this one. We use without sample, and this one look at number one. And how about with the sample? This below that this is a display that almost MC absent cell. If we add sample fraction, and this is without sample. I think didn't show that. Didn't show it about positive control.
Okay, Mrs. Niati, can you stop screen your slide? How is Lucy? Miss Lucy, are you there?
Yes.
I mean the question from Miss Lucy about normal cell line is mean like not cancer cell line, just like fibroblasts. Have you done it? Have you tested your compound with the normal cell line instead of using cancer cell line?
to answer this, Luciana, this one, maybe
yes.
we collaborate with Anas Fitria Devi and Nina. They did this research about how about our extract. They have positive anti-cancer activity or no, or didn't have anti-cancer, and we got depression active anti-cancer activity. Maybe.
Okay, thank you so much, Mrs. Osniati. Maybe you can discuss detail with your results with Lucia, because here there is no response from Miss Lucy. So next question from Ahmad Fatoni, also from to Mrs. Husniati. Do you have any data for your study related to anti-cancer activity of Robusta extract, probably caffeine contents?
Thank you for Pak Ahmad Fatoni. Our research didn't have caffeine, didn't have anti-cancer activity for caffeine, because we concern for polyphenol compound, especially CTA and their fraction. They have antioxidant activity, but caffeine didn't have any, antioxidant. And then we, this is answered about we didn't concern about caffeine. Thank you.
How is it, Dr. Ahmad Fatoni?
Okay, thank you for your answer. But as we know, the caffeine also have anti-cancer activity in other study. Maybe we can, the caffeine also maybe contribute to anti-cancer in your study. Okay, thank you very much for your answer.
Maybe different anti-cancer, different breast cancer, maybe the other cell line.
Different cell line, you mean like that, right?
Yeah.
Oh, okay. Thank you. Okay, thank you so much, Dr. Ahmad Fatoni and Mrs. Osniati. I think we still have 10 minutes more for discussion. If you have the question, you can ask our presenter today. Okay, can I ask one question to Mr. Jenny, because actually this research it's quite familiar with me, because I did this research when I was in undergraduate student long time ago. End up of this research is you would like to replace the chemical compounds, just like abate, something like that, with your natural lymphocyte, right? So, in your opinion, how much the concentration of this essential oil is required if you want to apply in the big container, something like that? Because in your study you only use petri dish as the model of the lymphocyte activities. Dr. Renhard Jenny, are you there?
Okay, thank you, Mr. Moderator. Larva.
No, in your opinion, how much concentration is required if you would like to apply in the big container? just like in the drum or something like that. Because in your study you use only the small space, right?
Yang bagus.
Ya, untuk menentukan warnanya.
Okay. Concentration inhibits concentration mortalization in 25 milligram per liter. Yeah. And so 10 and 10 larva visit in research, moderator.
It means your result, your essential is equivalent with the chemical compound abate, something like that.
Sama enggak itunya sama.
Comparison and
okay. Sit in. I'm sorry. Segar and kering in the activity in drying leaves in activity. Mr. Moderator.
All right, thank you.
Okay, thank you very much.
Okay, I think we have 10 more minutes for Q&A session. If you have any question to ask. Well, any more question? Okay, if there is no question, we can stop our discussion, our parallel session today. Please be informed you again. Tomorrow, don't forget to join us again in our second day of ISAC Symposium 2020. I think that's all. Thank you very much for your attention, your contribution, and your time for joining us in our symposium. Thank you very much for presenter to present your research to us. And I think that's all. We can leave. I ask moderator, so you can leave this meeting and don't forget to join us again tomorrow. See you and Assalamualaikum warahmatullahi wabarakatuh and stay healthy.
Waalaikumsalam warahmatullahi wabarakatuh.
Sukses selalu, Pak.
Thank you. Yep.
Apa aja nih?
Halo Pak Gunawan, sukses selalu.
3, 2, 3, 4, 5, 3, 36 mana? Ini 36 siapa? Muhammad Tarif.
you.